Hey everyone, welcome to my live stream. Today, we're going to be doing a number of things. First things first is I have a new cat. She's here with me now. She's a little wild beast. And she can't be tamed by any man, woman, or child. No clue on the name, although I think she has like this little weird meow. You can kind of hear it there. Shut up. Shut up, kitty. Shut up, kitty. You're not even a real kitty. Yeah. So what I thought we'd do today is I was gonna do like an hour each in like 5 different Harvard courses. So I didn't go to Harvard, and probably most people watching didn't either, just because not that many people go to the school in the first place. So it's gonna be fun to go through each textbook that you can just download online or whatever, purchase, and get the same Harvard education for free. This cat, though, is a real troublemaker. Come here, kitty. Yeah, yeah, yeah. Yeah, yeah, shut up. Come on, kitty. What? Now you're not a real kitty. Yeah, so what? I went to jail. So what? Oh, okay, you're better than me? Is that what you're saying? You're 9 weeks old? You're already better than me? Oh, please. Oh, please. Come on. Come up here. Yeah, come up here. Say something in Albanian. This is my beautiful maca. Maca bukur. Maca bukur. Shumë bukur. Right? Right. What? What is that sound? What is that sound? What is that sound? I love you, kitty. I love you. I love you, kitty. Yes, I do. Yes, I do. Yes, I do. Okay. Let's get to work. All right, so I've got these textbooks here. I swear to God, kitty, I'll snip your tail off. I snipped that tail right off. Or snip it right off. Let's start with something easy. What sound is that? That's not a meow. There's not even a meow. That's a garbage cat sound right there. Somebody gotta teach you how to meow. All right, let's get to work. Enough of this silly feline. All right, so, ow! This fucking cat just scratches me with no problem. All right, so this is called Essential Cell Biology, evidently. And we're gonna read that for an hour while this cat meows and meows and meows. You gotta be kidding me. You got to be kidding me. What is that? Is that a meow? Is that a meow? Yeah? Oh yeah? You've been scratching me too much, kitty. You know that? You've been scratching me too much. You don't know your own strength. Yeah. You've been scratching me way too much.
Hello, hello. How's the cat?
It's alright, I love you. It's alright, I love you. She's good. She's good. She's very small. S-M-O-L.
What did you name her?
I haven't named her yet, but she's very, very small. I can hold her in one hand.
I haven't even seen her yet. Do you have her up on stream?
Yeah, I had her on stream. She's going to be with me for the next 20 years, so... Alright,
God bless.
I'm doing this Harvard thing. Let's go through this 5th edition, Essential Cell Biology. This is Albert's. We're going to do an hour or so a piece in an entire Harvard undergraduate curriculum. So UCSF. Published by Norton, came out in 2019. What's up?
Bio lesson here?
Yeah, just going through an entire Harvard undergraduate coursework, at least in STEM. I'm going to probably skip some of the humanities stuff. We could do that if people want, but we'll just try STEM.
Awesome.
All right, this is the preface. Nobel Prize winning physicist Richard Feynman once noted that nature has a far, far better imagination than our own. Few things in the universe illustrate this observation better than the cell. A tiny sack of molecules capable of self-replication, this marvelous structure constitutes the fundamental building block of life. We are made of cells. Cells provide all the nutrients we consume, and the continuous activity of cells makes our planet habitable. Let me zoom in a little. Try to gauge how much I can see. All right. To understand ourselves and the world of which we are a part, we need to know something of the life of cells. Armed with such knowledge, we as citizens and stewards of the global community, speak for yourself, will be better equipped to make well-informed decisions about increasingly sophisticated issues from climate change and food security to biomedical technologies and emerging epidemics. In Essential Cell Biology, we introduce readers to the fundamentals of cell biology. The 5th edition introduces powerful new techniques that allow us to examine cells and their components with unprecedented precision, such as super-resolution fluorescence microscopy and cryo-electron microscopy, as well as the latest methods for DNA sequencing and gene editing. We discuss new thinking about how cells organize and encourage the chemical reactions that make life possible, and we review recent insights into human origins and genetics. With each edition of ECB, Essential Cell Biology, its authors re-experience the joy of learning something new and surprising about cells. We're also reminded of how much we still don't know. Many of the fascinating questions, the most fascinating questions in cell biology remain unanswered. How did cells arise on the early Earth, multiplying and diversifying through billions of years of evolution to fill every possible niche, from steaming vents in the ocean floor to frozen mountaintops, and, in doing so, transform our planet's entire environment? How is it possible for billions of cells to seamlessly cooperate and form large multicellular organisms like ourselves? These are among the many challenges that remain for the next generation of cell biologists, some of whom will begin a wonderful lifelong journey with this textbook. Readers interested in learning how scientific inquisitiveness can fuel breakthroughs in our understanding of cell biology will enjoy the stories of discovery presented in each chapter's How We Know feature. Packed with experimental data and design, these narratives illustrate how biologists tackle important questions and how experimental results shape future ideas. In this edition, a new How We Know recounts the discoveries that first reveal how cells transform the energy locked in food molecules into the forms used to power the metabolic reactions on which life depends. As in previous editions, the questions in the margins at the end of the chapter not only test comprehension, but also encourage careful thought and the application of newly acquired information to a broader biological context. Some of these questions have more than one valid answer, and others invite speculation. Answers to all the questions are included at the back of the book, and many provide additional information or alternative perspective on material presented in the main text. More than 160 video clips, animations, atomic structures, and high-resolution micrographs complement the book and are available online. The movies are correlated with each chapter, and callouts are highlighted in color. This supplemental material, let's get my teaching assistant here. This supplemental material, created to clarify complex and critical concepts, highlights the intrinsic beauty of living cells. For those who wish to probe even more deeply, Molecular Biology of the Cell, now in its 6th edition, offers a detailed account of the life of the cell. In addition, Molecular Biology of the Cell, 6th edition, A Problems Approach by Wilson and Hunt, provides a goldmine of thought-provoking questions at all levels of difficulty. We have drawn upon this tour de force of experimental reasoning for some of the questions in essential cell biology, and we are very grateful to its authors. Every chapter of ECB is the product of a communal effort. Both text and figures are revised and refined as drafts circulated from one author to another, many times over and back again. The numerous other individuals who have helped bring this project to fruition are credited in the acknowledgments that follow. Despite our best efforts, it is inevitable that errors have crept into the book, and we encourage eagle-eyed readers who find mistakes to let us know so that we can correct them in the next printing. Here are all the acknowledgments, and we will move on to chapter 1. Let's look at the authors very briefly. Bruce Alberts received his PhD from Harvard and is professor in the Department of Biochemistry and Biophysics at UCSF, editor-in-chief of Science, wow, from 2008 to 2013. That's one of my favorite journals. And served as the president of the USNAS from 1993 to 2005. Wow, very cool. I know. Yes, we're going to talk about that too. Yes, we're doing linear algebra next. Shut up, kitty. Wait your turn. I know you already know biology. Yeah. Well, if you could teach it better, come on up here and do it. Psh. Shut up. That's not even a real meow. You don't even know how to meow. Come here. She just wants to play. She wants attention. Let's play. You want to play? Let's play for 1 minute. We'll take a 1 minute break to study the biology of the wild beast cat. Yeah? Yeah? What? Yeah. You want a treat? Yeah. I'll get you a treat. Hold on. Let's get you some treats. You want some treats? Fine. F you too. All right, she's licking her mouth. That means she's hungry. She wants some treats. There we go. Now you'll be quiet. All right. Chapter 1. Cells, the fundamental units of life. It's true. What does it mean to be living? Petunias, people, and pond scum are all alive. Stones, sand, and summer breezes are not. But what are the fundamental properties that characterize living things and distinguish them from non-living matter? The answer hinges on a basic fact that is taken for granted now, but marked a revolution in thinking when first established more than 175 years ago. All living things or organisms are built from cells, small, membrane-enclosed units filled with a concentrated aqueous solution of chemicals and endowed with the extraordinary ability to create copies of themselves by growing and then dividing in two. The simplest forms of life are solitary cells. Higher organisms, including ourselves, are communities of cells derived by growth and division from a single founder cell. Every animal or plant is a vast colony of individual cells, each of which performs a specialized function that is integrated by intricate systems of cell-to-cell communication. Just so you know, we're going to talk about unity and diversity of cells, cells under the microscope, the prokaryotic cell, the eukaryotic cell, and model organisms. Cells, therefore, are the fundamental units of life. Thus, it is to cell biology, the study of cells and their structure, function, and behavior, that we look for an answer to the question of what life is and how it works. With a deeper understanding of cells, we can begin to tackle the grand historical problems of life on Earth, its mysterious origins, its stunning diversity produced by billions of years of evolution, and its invasion of every conceivable habitat on the planet. At the same time, cell biology can provide us with answers to the questions we have about ourselves. Where did we come from? How do we develop from a single fertilized egg cell? How is each of us similar to, yet different from, everyone else on Earth? Why do we get sick, grow old, and die? In this chapter, we introduce the concept of cells, what they are, where they come from, and how we have learned so much about them. We begin by looking at the great variety of forms that cells can adopt, and we take a preliminary glimpse at the cellular machinery or chemical machinery that all cells have in common. We then consider how cells are made visible under the microscope, and what we see when we peer inside them. Finally, we discuss how we can exploit the similarities of living things to achieve a coherent understanding of all forms of life on Earth, from the tiniest bacterium to the mightiest oak and the mightiest cat, right? If you're just joining us, we're doing 5 different Harvard courses this morning. And we have a very loud guest lecturer who wants to be heard every moment. I'm sorry about that. A big part of my live streams before I took a 4 or 5 year vacation was education. And I thought it'd be fun to download 5 Harvard textbooks that your average Harvard student would be studying and read those textbooks. And some people may want to listen in along. Some people may have learned this a long time ago. Some people may not give a crap and just are here for the cat or my beautiful face. But regardless, that's the program for today. Biologists, so let's talk about the unity and diversity of cells. Biologists estimate that there may be up to 100 million distinct species of living things on our planet. Organisms as different as a dolphin and a rose, or a bacterium and a butterfly. Cells, too, differ vastly in form and function. Animal cells differ from those in a plant, and even cells within a single multicellular organism can differ wildly in appearance and activity. Yet, despite these differences, all cells share a fundamental chemistry and other common features. In this section, we take stock of some of the similarities and differences among cells, and we discuss how all present-day cells appear to have evolved from a common ancestor. Yeah? Cells vary enormously in appearance and function. When comparing one cell to another, one of the most obvious places to start with is size. A bacterial cell, say a lactobacillus, in a piece of cheese, is a few micrometers, or micro-m. This is the Greek letter mu. If you haven't seen it before, I have my Wacom tablet. Maybe I should plug it in. I don't know where the stylus is, though. The stylus would probably help, wouldn't it? I'll have to find that. Shut up, kitty. Yeah, I think this isn't too useful without the stylus. Well, anyway, I was going to just sort of point out, we all know what 1 meter is, right? How long is a meter? around 3 feet. 3 feet, right? It's like 3.2 feet exactly, I think. So it's almost like a yard. Let's see. 1 meter, 2 feet. 3.28 actually. So it's more like 3.3. 3.28084. All right, so 1 millimeter is 1,000th of that. So we know how big a millimeter is, right? It's very, very thin. Extremely small distance. Centimeter is about the length of a finger knuckle. So a millimeter is 1/10 of a centimeter. So a micrometer is 1/1000th of a millimeter, and 1/1,000,000th of a meter. Is a micrometer visible? This is supposed to be micrometer. No, it's not visible. It's
I wouldn't think it would be.
just below the threshold of, probably we lose the ability to discern maybe around like 100 micrometers. So 1 micrometer is too small. So that's about 25 times smaller than the width of a human hair. So that's the diameter maybe, or length of a bacterial cell. So not quite so microscopic. It's microscopic, but it's not like it's so small it's almost, impossible to see. It's definitely not within the ability for us to discern, but it's not like it's that far away. It's not like it's an atom or something. At the other extreme, a frog egg, which is also a single cell, has a diameter of about 1 millimeter, which is technically visible. If we scale them up to make the lactobacillus the size of a person, the frog egg would be half a mile high. Cells vary just as widely in their shape. A typical nerve cell in your brain, for example, is enormously extended. It sends out its electrical signals along a single fine protrusion called an axon that is 10,000 times longer than it is thick. So it's very long, like a piece of string almost. And the cell receives signals from other nerve cells through a collection of shorter extensions that sprout from its body like the branches of a tree. A pond-dwelling paramecium, on the other hand, is shaped like a submarine and is covered with thousands of cilia, hair-like projections whose sinuous, coordinated beating sweeps the cell forward, rotating as it goes. A cell in the surface layer of a plant is squat and immobile, surrounded by a rigid box of cellulose with an outer waterproof covering of wax. A macrophage in the body of an animal, by contrast, crawls through tissues, constantly pouring itself into new shapes as it searches for it and engulfs debris, foreign microorganisms, and dead or dying cells. A fission yeast is shaped like a rod, whereas a budding yeast is delightfully spherical, and so on. Cells are enormously diverse in their chemical requirements. Some require oxygen to live, for others the gas is deadly. Some cells consume little more than carbon dioxide, sunlight, and water as their raw materials. Others need a complex mixture of molecules produced by other cells. These differences in size, shape, and chemical requirements often reflect differences in cell function. Some cells are specialized factories for the production of particular substances such as hormones, starch, fat, latex, or pigments. Others, like muscle cells, are engines that burn fuel to do mechanical work. Still others are electricity generators, like the modified muscle cells in the electric eel. I don't know if I'd go to Harvard to do a postgrad, but some modifications specialize a cell so much that the cell ceases to proliferate, thus producing no descendants. Such specialization would be senseless for a cell that lived a solitary life. In a multicellular organism, however, there is a division of labor among cells, allowing some cells to become specialized to an extreme degree for particular tasks, and leaving them dependent on their fellow cells for many basic requirements. Even the most basic need of all, that of passing on the genetic instructions of the organism to the next generation, is delegated to specialists, the egg and the sperm. Living cells have a similar basic chemistry. Despite the extraordinary diversity of plants and animals, people have recognized from time immemorial that these organisms have something in common, something that entitles them all to be called living things. But while it seemed easy enough to recognize life, it was remarkably difficult to say in what sense all living things were alike. Textbooks had to settle for defining life in abstract general terms related to growth, reproduction, and an ability to actively alter their behavior in response to the environment. Let's actually look here at question 1.1. Life is easy to recognize but difficult to define. According to one popular biology text, living things are 1, highly organized compared to natural inanimate objects. 2, they display homeostasis, maintaining a relatively constant internal environment. 3, they reproduce themselves. 4, they grow and develop from simple beginnings. 5, they take energy and matter from the environment and transform it. 6, they respond to stimuli. 7, they show adaptation to their environment. Score of person, vacuum cleaner, and a potato with respect to these characteristics. Okay. The discoveries of biochemists and molecular biologists have provided an elegant solution to this awkward situation. Although the cells of all living things are enormously varied when viewed from the outside, they are fundamentally similar inside. We now know that cells resemble one another to an astonishing degree in the details of their chemistry. They are composed of the same sorts of molecules, which participate in the same types of chemical reactions, discussed in Chapter 2. In all organisms, genetic information, in the form of genes, is carried in DNA molecules. This information is written in the same chemical code, constructed out of the same chemical building blocks, interpreted by essentially the same chemical machinery, and replicated in the same way when a cell or organism reproduces. Let's take a break to look at figure 1.1. So we're looking at 100 micrometer resolution here. Figure 1.1, cells come in a variety of shapes and sizes. Note the very different scales in these micrographs. This one's 25, 25, 5, and 3. Drawing of a single nerve cell from a mammalian brain. This is here. A. This cell has a single unbranched extension, the axon. You can see it there. Projecting towards the top of the image, though through which it sends electrical signals to other nerve cells, and possesses a huge branching tree of projections, these are called dendrites, through which it receives signals from as many as 100,000 other nerve cells. And again, notice the scale here. This is 100 micrometers, almost visible amount, or length, I should say. This is a paramecium, a commonly studied protozoan, a single giant cell, swims by means of the beating cilia that covers its surface. The surface of a snapdragon flower petal displays an orderly arrangement of packed cells. D is a macrophage, spreading itself out as it patrols animal tissues in search of invading organisms. And A, a fission yeast, is also caught in the act of dividing into two. The medial septum, stained red, is forming a wall between the two nuclei, also stained red, that have separated into two daughter cells. The cell membranes are stained green. All right. In every cell, long polymer chains of DNA are made from the same set of 4 monomers called nucleotides. Very important, nucleotides. This is where we get mRNA, which we'll talk about in a second. Strung together in different sequences, like the letters of an alphabet. The information encoded in these DNA molecules is read out, or transcribed, very important, transcribed, into a related set of polynucleotides called RNA. Although some of these RNA molecules have their own regulatory, structural, or chemical activities, most are translated. So we have transcribed and translated. The DNA gets transcribed into RNA, and RNA gets translated into a different type of polymer called a protein. So a polymer is just any molecule with repeating units. Any molecule with repeating units. That includes plastics or polymers, but also biomolecular molecules too. This flow of information from RNA to DNA, from DNA to RNA to protein is so fundamental to life that it is referred to as the central dogma of biology. And here's a look at it from this side. DNA, RNA, protein. DNA synthesis replicates. That's not too important right now. That's when cells divide and things like that. But RNA synthesis is when we go from DNA to RNA. These are both nucleotides. There's some subtle differences that make RNA, RNA, and DNA, DNA. But either way, from this RNA template, we can translate this into proteins, and these proteins are made of amino acids. So that's the central dogma. The appearance and behavior of a cell are dictated largely by its protein molecules, which serve as structural supports, chemical catalysts, molecular motors, and much more. Proteins are built from amino acids. There's one thing you need to know in biology, this is it. And all organisms use the same set of 20 amino acids. We have 26 letters in English, but God has 20 amino acids to make their proteins. But the amino acids are linked in different sequences. Just the same way we can make a lot of different words with 20 amino acids, we can make a lot of different proteins with, we can make a lot of words with 26 alphabet letters, we can make a lot of different proteins with 20 amino acids. But the amino acids are linked in different sequences, giving each type of protein molecule a different three-dimensional shape or conformation. So even the change of one amino acid in the wrong place can create a disastrous situation. Let me show you. We're going to look up some protein sequences using this tool from the government. Thank you, government. Well, let's do a different. Let's go here. So this is dystrophin, which is the gene for the protein that, when mutated, causes muscular dystrophy. You can see it's in all different types of organisms. So I'm just going to type in dystrophin homo to get Homo sapiens. And if I look at this gene and I want to know the sequence, I can click to get the sequence. I haven't used this website in years. It should be here somewhere. Here it is. So this is the DMD, this is a gene that's on the X chromosome. That's why it's an X-linked disorder. So only boys get muscular dystrophy or Duchenne muscular dystrophy because girls have two copies of the gene. They have an X copy and a Y copy, or an X copy and an X copy. Boys only have an X copy, one copy, because they have a Y chromosome. So if you mutate the one copy, there's no backup copy. Females have a backup copy. So here's the gene sequence, and you can see it's a very intricate detail, but and it goes on and on. In fact, dystrophin is one of the biggest genes in the human body. One misspelling of this, just one of these, right? How many are there? What does it look like? Look at the bottom of my screen. You can see the scrolling is going up very slowly. What would you guess, 1 million? It's enormously large, right?
Even
2 million. So there's all kinds of tools in here to go through it, but it's an enormous protein. And there's all kinds of different ways you can look at these. And again, if you want to do this yourself, just go to the NCBI. But yeah, I think that you can get a sense for just, this is a very large protein, or gene I should say, but there are many genes that are smaller. There's most genes in fact are a lot smaller than this one. But you can see that this gene that encodes for this important protein called dystrophin, it's such an important protein when it comes to muscle, that a single wrong nucleotide can cause death, unfortunately, in people. So let's keep going. All right, so amino acids are linked in different sequences, giving each type of protein molecule a different three-dimensional shape or conformation, just as different sequences of letters spell different words. In this way, the same basic biochemical machinery has served to generate the whole gamut of life on Earth. Right, in all living cells, genetic information flows from DNA to RNA and from RNA to protein. That's transcription from DNA to RNA and translation from RNA to protein, an arrangement known as the central dogma. The sequence of nucleotides in a particular segment of DNA, which is a gene, is transcribed into an RNA molecule, which can then be translated into the linear sequence of amino acids of a protein. Only a small part of the gene, the RNA and protein, is shown. All right, living cells are self-replicating collections of catalysts. That's a great way to put it. One of the most commonly cited properties of living things is their ability to reproduce. For cells, the process involves duplicating their genetic material and other components and then dividing into two, producing a pair of daughter cells that are themselves capable of undergoing the same cycle of replication, sort of an exponential growth. It is the special relationship between DNA, RNA, and proteins, as outlined in the central dogma, that make this self-replication possible. DNA encodes information that ultimately directs the assembly of proteins. The sequences of nucleotides in a molecule of DNA dictates the sequence of amino acids in a protein. All right, proteins in turn catalyze the replication of DNA and the transcription of RNA. And they participate in the translation of RNA into proteins. This feedback loop between proteins and polynucleotides underlies the self-reproducing behavior of living things. So this is interesting, and there's one author named Douglas Hofstadter, I think, who wrote a book called Gödel, Escher, Bach, which is a very influential book. I recommend the book. It's good, not great. And anyway, the point that he makes is, and many people have observed this, is that when you look at, hold on, my cat messed up a lot of my infrastructure here. When you look at DNA, it's self-referential. What does that mean? This central dogma is called dogma for a reason. Dogma is usually used like kind of an insulting word, right? Well, the reason this is kind of interesting is that this is the central dogma. But the reality is a bit different. There's a lot of other interactions that are occurring. Where I won't talk too much about those just yet. That's what this whole book is about. But what I want you to understand is that, sorry, I don't know what my computer's doing right now. What I want you to understand is basically this idea that when some of these proteins, right, are related to helping DNA replicate and maintaining their status. So there's this feedback loop that's very interesting, right? There are proteins that help DNA replicate. And those proteins are created by the very DNA. So some people call this recursive in computer science. There's a lot of concepts like this in art and music, which is why it's called Gödel, Escher, Bach. And Gödel's incompleteness theorem has a recursive idea in it. So and Hofstadter's general theme of the book is that this is where like all complexity and intelligence and beauty lies in this self-referencing sort of strange loop is what he calls them. He even has something called I Am a Strange Loop. I think it's a book, and maybe even became a musical, I don't know. But anyway, it's an interesting book. But the point is, you really can't have DNA without the proteins that allow the DNA to have its elegant song and dance of this is some DNA, of basically chaperoning it to divide and separate and then rewind again and come apart and then having the cells kind of divide. This is two cells dividing. starts off as one cell, and you can see them kind of budding out, and then they separate. Proteins are required to do this, but wait a second, the data for the proteins are stored in the DNA. The RNA is kind of like this intermediate. So, it's sort of like this very funny thing. Well, if DNA comes from proteins and proteins come from DNA, how does this all work? And it's kind of amazing to think about. And I think the thing about Hofstadter, though, is a good scientist and everything, but the fact that this like blew his mind is probably a little like memey, and it kind of reminds me of like maybe the stereotypical pothead or something, saying, "Whoa, bro, this is crazy." The reality is it's very explainable and not that deep as maybe the connections that he's drawing that he thinks they are. So anyway, that's just my 2 cents. So let's keep going here. DNA encodes information that ultimately directs the assembly of proteins. The sequences of nucleotides in a molecule of DNA dictates the sequence of amino acids in a protein. Proteins, in turn, catalyze the replication of DNA and the transcription of RNA, and they participate in the translation of RNA to proteins. This feedback loop between proteins and polynucleotides underlies the self-reproducing behavior of living things. We discuss in complex interdependence between these 3 actors in detail in chapters 5 through 8. In addition to their roles in polynucleotide and protein synthesis, remember, polynucleotide just means DNA or RNA, a lot of nucleotides. Proteins also catalyze the many other chemical reactions that keep the self-replicating system shown in 1.4 running. So let's see what 1.3 is here. These are all living organisms are constructed from cells. This is a colony of bacteria, again, 2 micrometers, not quite visible, and bacteria could be good or bad. A butterfly, a rose, and a dolphin are all made of cells that have a fundamentally similar chemistry, which is really amazing that the DNA, if you compare the DNA amongst these 4 organisms, they're very, 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 very similar. Which surprised me when I learned that the DNA contents of a butterfly and a dolphin and a rose, you can imagine these things are so different. Most of the machinery is the same. Most of the way that we harvest energy and things like that has not changed in a billion years, which is really kind of amazing if you think about it. Or maybe 500 million years, better way to look at it, or 100 million years. All right. A living cell can break down nutrients and use the products to make both the building blocks needed to produce polynucleotides, DNA and RNA, proteins, and other cell constituents. What's in a cell? We'll find out soon. And to generate the energy needed to power these biosynthetic processes. So it's using biology to make things. We discussed these vital metabolic reactions in detail in chapter 3 and 13. This word metabolic confuses a lot of people, but basically metabolic is just any kind of biosynthetic process. And we sometimes say metabolism. And some people say, well, I have a fast metabolism or slow metabolism, and I think that's, to me, is often in their head. But the point is, any transformational reaction from one entity to another can be thought of as a metabolic reaction. Let's look at figure 1.4. Life is an autocatalytic process. So auto means automatic or will occur on its own. Catalytic means something happening or something being created or transformed. DNA and RNA provide the sequence information, the green arrows, that is used to produce proteins and to copy themselves. Proteins, in turn, provide the catalytic activity needed to synthesize DNA, RNA, and themselves. So it's amazing that the proteins are kind of machines that make all this happen, and the DNA is the information. It's almost like the software and hardware to some extent. Together, these feedback loops create the self-replicating system that endows living cells with their ability to reproduce. And as you study this, I think you'll find it to be rather amazing how you can have a protein that's sort of guiding or even dozens of proteins or hundreds of proteins that are guiding and shaping the DNA and RNA so that the DNA and RNA can transform into more protein. So the very proteins that are guiding the DNA and RNA are sequenced from DNA and RNA. So there's no top level, if you will. It all starts sort of at DNA, I suppose, but there's still kind of this very intricate meta transformation. You don't usually see that in things like computer programming, where you sort of have a core level of DNA, and typically, I suppose well, maybe it is somewhat similar. I don't know. It's something very interesting to ponder and think about. Only living cells can perform these astonishing feats of self-replication. Viruses also contain information in the form of DNA and RNA, but they do not have the ability to reproduce by themselves. Instead, they parasitize the reproductive machinery, or they take over the reproductive machinery of cells that they invade to make copies of themselves. This is like the COVID virus will take over some of your cells to make a copy of itself. Thus, viruses are not truly considered living. They're merely chemical zombies, inert and inactive outside of their host cells, but able to exert a malign or malignant control once they gain entry. We review viruses in Chapter 9. Yeah, if you look at my DNA, there's loyalty instead of A, C, T, and G, but that's another topic. Scientists are very confused by that. Thank you, Kendrick Lamar. All living cells have apparently evolved from the same ancestral cell. This is a very, very unusual idea. All living cells have evolved from the same ancestral cell. When a cell replicates its DNA in preparation for cell division, the copying is not always perfect. I think this is probably thing number 2 to really understand in biology. This will help you understand a lot. When a cell replicates its DNA in preparation for cell division, the copying is not always perfect. Well, why? Well, we just looked at the NCBI, right? NCBI, National Center for Biotechnology Information. We can go to the genome domain or gene domain. We can type in, I don't know, insulin. Do you want to type in insulin? Any other protein some people care about? There's insulin, there's the insulin receptor, there's the insulin precursor. I'm going to type in insulin homo, again for the species, but I could do mus musculus, which is just a mouse, and we could see the insulin gene in mice. We can look at KRAS, which is a very unusual protein. Insulin is most definitely a protein. It's made from an insulin precursor. So let's look at, and then it has a receptor, and there's IGF, which is not insulin, but related. Here's an insulin-degrading protein. You can see there's dozens of proteins related to insulin, but the big one is called pre-pro-insulin, I think, or pre-insulin. I think it might be. But a lot of hormones have a pre-pro kind of concept within different, let's see. There's pre-insulin, pro-insulin, there's pre-pro in the elephant shark. Well, this is plain old insulin on chromosome 10. So maybe insulin doesn't get chopped up and metabolized. Anyway, let's go to the genome sequence. Here's some of the ways to look at the sequence, but I'm just going to go to the basic FASTA, which is just the letters, because I think that's better. There it is. It's really small relative to dystrophin, right? Dystrophin was like millions of base pairs. These are each called base pairs. And you can see there's 4 letters, A, C, T, and G. So anyway, insulin is really, really short. And we're going to learn in different chapters that these three nucleotides each code for a different amino acid. And this is around 44 amino acids, if I'm not mistaken. So we're going to determine and learn a bit about this down the road. Why did I mention this? Because when we looked at dystrophin, which was so large, it made sense to me, at least, that there was possible that you couldn't necessarily replicate this too easily, right? That maybe something would go wrong. It was millions of base pairs. I can't even scroll down fast enough to get to the bottom. This is maybe the fastest I can scroll, and it would still take me about a minute to scroll down to the bottom. So how could it be possible that this gets copied perfectly every single time? It would have to be a really good copy machine, right? We have this copy machine inside of our body that is this good. And when we think about computers, we think about copying and pasting. When you copy and paste something, it's not like one thing in the copy-paste will go wrong, right? It's not like one letter will accidentally get changed. Well, that's because our computer software is a little bit different from our body software. In our body software, every now and then, one of these will, instead of an A, T, C, or G, it might go to an A, C, T, or G in a different order. And that's all it takes for there to be a so-called mutation. And you can see just how long this was, right? This is so enormous. How do you get it exactly the same every time? That's sometimes called fidelity. And we use that term in music or audio sometimes, hi-fi, lo-fi. So high fidelity is a perfect copy. Well, on occasion, the instructions are corrupted by mutations that change the sequences of nucleotides in the DNA. For this reason, daughter cells are not necessarily exact replicas of their parents. Mutations can create offspring that are changed for the worse, in that they are less able to survive or reproduce, or changed for the better, in that they will be better able to survive or reproduce, or change in a neutral way, in that they are genetically different but equally viable. The struggle for survival eliminates the first, favors the second, and tolerates the third. The genes of the next generation will be the genes of the survivors. That's probably the next most important thing to understand in biology. Central dogma: cell replication does not result in a perfect copy, and the genes that survive are the genes of the survivors. Now, we notice that there are still genetic diseases despite this, right? And that's because the window for reproduction and survival are two different things. You can have Duchenne muscular dystrophy, which has a lifespan of around 20 years, and still reproduce. You can have a lifespan of 40 years with Huntington's disease and still reproduce. So it's not a perfect sort of one-to-one ratio. If you have a fatal disease that results in death before the age of 5, then the genes of the next generation will be the genes of the survivors and not the genes of that patient, for instance. So anyway, I'd encourage you to think about this a little bit, that it seems to be that most mutations are actually ones that change you for the worse. Having said that, the only ones that really carry on over long periods of time are the ones that change you for the better. So it's a very interesting sort of concept. For many organisms, the pattern of heredity may be complicated by sexual reproduction, in which two cells of the same species fuse, pooling their DNA. The genetic cards are then shuffled, redealt, and distributed in new combinations to the next generation, to be tested again for their ability to promote survival and reproduction. One way to look about or think about DNA is basically DNA is just a game. And the game is, can you reproduce and send your DNA? Here's some DNA here. Here's some DNA here. And the game is, can this DNA reproduce better or more than this DNA? And that's all there is to life. is who can reproduce the most. And if you think about it that way, it's a little bit of a cold world, but it is definitely the kind of dynamic that is sort of what gives rise to life. This DNA is competing for resources. The machines that it makes, the DNA makes proteins. So those proteins are molecular machines. It's trying to make the best molecular machines that will reproduce itself. If it can't do that, it'll die. So this DNA might make a protein organism of a cat. Well, this one made a cat, but a cat with no claws. Well, this one's going to survive and reproduce, more likely at least, than this one. And maybe that's why cats have very sharp claws. And so forth and so forth. And all the way up to perhaps even consciousness. And there's a lot of debate as to whether consciousness, what we seem to exhibit, is a byproduct of evolution or a direct cause of evolution. And we don't know. But it's a good question. All right. These simple principles of genetic change and selection, applied repeatedly over billions of cell generations, are the basis of evolution, the process by which living species become gradually modified and adapted to their environment in more and more sophisticated ways. Evolution offers a startling but compelling explanation of why present-day cells are so similar in their fundamentals. This is probably the best proof. There's a lot of proof of evolution, and a lot of it is just common sense. But one very simple proof is that if you look at the DNA of early organisms, you can actually see and chart on a tree of life, or a more complex chart, exactly when certain things happened. For example, one protein I've studied very closely is called oxytocin. Oxytocin is a hormone, sometimes called the love hormone, jokingly. It's an octamer, I believe, or nonomer. It started off as an octamer, and it actually evolved from a different hormone called vasopressin. So fish had vasopressin before humans existed, and oxytocin was made just in a molecular accident by mutating by that low fidelity copying mechanism. Oxytocin just happened to be created. And over an evolutionary tree branch, you can see exactly when oxytocin showed up in evolution. So you can see when it showed up in organisms. And oxytocin is a fascinating little protein, sometimes called, like I said, the love hormone. You make oxytocin when you hug somebody or kiss somebody. Dogs make a stream of oxytocin when they see their owners. I don't like to use the word owner, but when they see their living companions. So it's very interesting how this protein works, but what's more interesting to me is that you can see what species have oxytocin and what species don't. Most unicellular organisms, like bacteria, don't have oxytocin. But what point did organisms grow enough to require it? And you can basically see that there's no doubt as to how this all came about. It came about by accidental replication of some of these proteins. There's no doubt whatsoever that this was a happy accident, and that humans are the production of lots of these little happy accidents. And over long enough periods of time, we can't think and conceptualize and contextualize just how long we're thinking about. We're thinking about millions and millions of years. Over enough generations, you'll see those differences. Mendel, who was a friar or a priest or something like that, right? Mendel, used pea pods, and he could see the evolution, or at least the hereditary traits, very quickly. It wasn't long before he realized, well, if you mate these two plants, this is what you get. And it isn't a surprise that over time you could influence the sort of drift of genes and species of these pea pods, because it was very quick it didn't take long to reproduce these. It takes a long time for humans to reproduce. So you don't see the changes immediately. But you could even think in your own head, if only these types of people reproduced, this would be the only progeny left, and then every now and then you'd have an accidental mutation that maybe would change hair color or skin color, and then you'd have to start all over again. So, it's just something to think about in terms of, it's pretty easy to understand from a common sense perspective. So anytime I hear somebody ask about the whether evolution is true or not, it's very frustrating to me because it's just something born of ignorance. All proteins evolved over time. That's correct. Gemini in chat is asking. That's exactly right. Proteins evolved just like species did. Very, very interesting way to look at it. All right. So repeated application of these billions of cell generations are the basis of evolution, the process by which living species became gradually modified and adapted to their environment in more and more sophisticated ways. Evolution offers a startling but compelling explanation of why present-day cells are so similar in their fundamentals. Our cells are the same as bird cells, and those are the same as bacterial cells, or they have a lot of the same characteristics in DNA. They all inherited their genetic instructions from the same common ancestral cell. It is estimated that this cell existed between 3.5 and 3.8 billion years ago, and we must suppose that it contained a prototype of the universal machinery of all life on Earth today. And again, that's debated quite a bit. It's not 100% clear how that happened or what it looked like. And we'll probably talk about that in a minute. But it is very interesting. And perhaps there are parallel, ancestors that created life. It's possible a lot of different things happen. So we don't have all the answers there quite yet. Through a very long process of mutation and natural selection, the descendants of this ancestral cell have gradually diverged to fill every habitat on Earth with organisms that exploit the potential of the machinery in a seemingly endless variety of ways. Well, I would dispute that a little bit. We actually have quite a similar need for things like ATP and other molecular nutrients. So I'd say that we're actually more similar with other organisms than maybe it's being said here. But regardless, mutations are mistakes in the DNA that change the genetic plan from that of the previous generation. Imagine a shoe factory. Would you expect mistakes in copying the shoe design to lead to improvements in the shoes produced? Explain your answer. Unintentional changes in copying shoe design. Well, I'd say most of those changes would not be good, right? But every now and then you might say, okay, let's use rubber instead. Let's use this or that, and maybe you would make a positive change, and then you'd say, okay, let's put that in our new factory plan. All right, genes provide instructions for the form, function, and behavior of cells and organisms. And we're going to probably wrap this up relatively soon and go to the next Harvard course. We're doing 5 different Harvard courses, undergrad courses today. Thanks for joining me. It's going to be like a bit of a marathon stream. And I'm feeling very energetic today, so I'm going to try to, do a marathon as much as I can. But who knows, I might tap out at some point. So genes provide instructions for the form, function, and behavior of cells and organisms. A cell's genome, this is what this word means, its entire sequence of nucleotides in an organism's DNA. Just how much is that? Let's look. Because we looked at dystrophin, but I want to look at the genome size of a human. How many nucleotides is that? 3 billion. 3 billion nucleotides. So the one we looked at, dystrophin, had 2 million nucleotides. So if you look at all of our genes, 3.2 billion nucleotides of DNA. And, in computers, they're binary, right? It's 1, 0, 1, 1, 0, et cetera, et cetera, et cetera, et cetera. That's the binary. But in DNA, it's ACTG, so it's quaternary. quaternary. There are 4 bases. And in numbers, we have 10 bases, right? 1, 2, 3, 4, etc. You could do pi. Etc. is the base 10 system. And in other computers, we have hexadecimal, right? My mouse is fritzing out here, but... And that's base 16. So our DNA is in base 4. There are 4 ways to distinguish. And of course, they're not little A's, T's, C's, and G's written in our DNA somewhere where you could look in a microscope and see, oh, look at that, that's an A. No, it's a molecule called adenine. These are different molecules. So they're not letters, so to speak, but their body kind of treats them the way we look at letters or codes. This is kind of a code. And the code gets more complex. When we look at amino acids, you're kind of talking about a base 20 system with amino acids. And amino acids have different letters like V or M or G and S. So you can go down Q, R. All these amino acids stand for these. All these are code for a specific amino acid. And again, if you look at a protein really closely, you don't see these letters. You see a little amino acid. So you can kind of make more complexity with more, a bigger kind of base system, right? To some extent, you can compress more information. But the beauty of binary code is how fast you can work with it. Well, DNA works very quickly too. So we'll kind of think about this sort of computer software version of biology, which is rapidly intersecting with real biology every day. And sometimes it's called bioinformatics. But I want you to keep that in the back of your mind, especially if you have a background in computers. So 3 billion base pairs. Well, I'm writing a program. Let's see, I wrote a program here earlier. Let's see how big this program was. Well, it's very, very small. It's 4,000 bytes. So 4,000 bytes, you know, in fact, if I think about, okay, well, how big is my entire... There used to be ways to do this. If I look at my entire programming system, how many bytes is that? I don't know, it's probably quite a lot. But is it 3 billion bytes? Well, what is 3 billion bytes? That's 3 gigabytes, right? So we have 3 gigabases. That's one way to look at it. So the one program I wrote to basically calculate duplications in the database is maybe like an insulin. It's small. But this entire folder, which is full of things called node modules, which take up a lot of space, basically other people's programs, they add up to quite a lot. And that's probably a good way to think about how large the human body is, or the genome. 3 billion base pairs, so 3 gigabytes of data. All right, so this is a genetic program that instructs a cell on how to behave. Sort of. That's not exactly what I would write, but that's their textbook, not mine. For the cells of plant and animal embryos, the genome directs the growth and development of an adult organism with hundreds of different cell types. Within an individual plant or animal, these cells can be extraordinarily varied, as we'll discuss in chapter 20. There's fat cells, skin cells, bone cells, nerve cells, and they seem dissimilar from each other, as any cells could be. Yet all these differentiated cell types, we're going to look at this term in really close detail, differentiated cell types, are generated during embryonic development from a single fertilized egg cell. That's kind of what's amazing, right? Is that even though all the cells are different, at the end, they kind of have a common ancestor even within one organism. And there's this old saying in biology, phylogeny or ontology recapitulates phylogeny. And the point is that just like we have a common ancestor that primordial cell, say P for that, and then say we have fish here, and then we have, I don't know, mammals here, and then finally humans. Just the same way we have that, we also have that in our own bodies, where we have, say, an egg cell, mesenchymal progenitor cells, then maybe we have a blood cell progenitor, like a hematopoietic stem cell, and then finally we'll have like an erythrocyte, which is a red blood cell. So we'll have that a weird descendant system even in our own, and of course lots of branches, even within our own bodies. So it's a very funny thing to see. And this old saying actually shows you that when the egg is dividing and growing, it's actually recapitulating some of the old evolution, that the fetus will kind of look like a fish and then kind of look like another organism over time during its embryonic development, which is kind of a little bit crazy and unusual. So we call these cells at the end of that chart, towards the bottom that I just showed you, differentiated. And there are cells that are less differentiated. And sometimes they're called pluripotent, or they have other words. But I want you to think about cells that are very differentiated versus cells that are a little less differentiated. And our organs have cells that are sometimes very differentiated and sometimes those that aren't. All right. All these different differentiated cell types are generated during embryonic development from a single fertilized egg cells, and they contain identical copies of the DNA of the species. So you would think that, let's look at that hematopoietic stem cell, and then you look at, let's say, a white blood cell, progenitor cell at least, and then you look at, let's say, a T cell. And then on the other side, you maybe look at a red blood cell. We've all seen red blood cells under microscopes, or what they kind of look like. And they're very simple cells that just have hemoglobin and carries oxygen. These cells are really different. But they all have the same DNA. Every single one of these cells, every single one has the same copy of DNA, the same master copy. So theoretically, why couldn't this cell be programmed to be this cell? What gave it its destiny to become a red blood cell? What gave this cell its destiny to become a T cell? And what gave this cell its destiny to stay as this kind of more neutral hematopoietic stem cell that could give rise to dozens of different types of cells? We used to think that these hematopoietic stem cells, which when people say they had a stem cell or a bone marrow transplant, this is what I'm talking about here. What made these bone marrow hematopoietic stem cells stay the way they are and always be ready to be a copy, a template for differentiation of other cell types? There used to be the thought, I was about to say, that people used to think that the hematopoietic stem cells could only differentiate into about 5 or 6 cell types. But now we're up to 20 or 30 different cell types in the blood system. So if you read an immunology textbook, it's rapidly becoming obsolete every year. You can barely keep up with that. We'll get to the four factors and Yamanaka factors later. So I do want to touch on advanced topics and beginner topics even within that first course. So just to get your mind ready and appetized to knowledge. I think that's one of the most important things in pedagogy, is making sure that you leave a couple morsels for what's next. And I think that excitement is what drives, to me, the motivation to learn. Otherwise, you lose that motivation. All right. Their varied characters stem from the way that individual cells use their genetic instructions. You can look at this gigantic dictionary or instruction manual, and you get a electronics, new piece of electronics. You might get an instruction manual that's 100 pages, and you don't read it. But or at least most people don't. I read every single page. But these cells kind of have that big, heavy instruction manual. It's tens of thousands of pages, and they go through it a page at a time to figure out I'm on page 83, or I'm on page 2 million, or I'm on page 75 million, and they look up what they're supposed to create. And each cell has a bit of a different situation. So different cells express different genes, and this is the key part. We have a lot of different genes. Anybody know how many? Those 3 billion nucleotides, how many genes are they composed of? Genes are like separate, and this thing keeps going and going. Genes, how many genes, human genes, are there? 20 to 25,000. I was going to say closer to 30,000, but regardless, those 3 billion nucleotides are split into 20,000. So what does that mean on average, right? On average, we got 3 billion. This is 3 million. So this is 3 billion. And then we have 25,000. So here's thousands. These will cancel out, right? And then we have 20. So these two will cancel out too. And 2 and 3 are pretty close, so we'll just fudge this to a 3. This is how I went to prison, by the way. I'll just sneak that, turn that 2 into a 3. And we know now that each, that was a joke, by the way, each, each gene has 100,000 nucleotides. I did not commit a crime, for what it's worth. That's what I was accused of. But it is what it is, and it's ancient history. 100,000 nucleotides per gene. So there's 25,000 genes, roughly. I think that number is higher than we think, but regardless, it's quibbling at this point. And there's 100,000-ish, looks like a little more, thanks to my little adjustment there, probably 150,000 nucleotides per gene. But we know that dystrophin, right, had 2 million. So just because there's another one called titinin, or titinin, that is, I think, the largest gene in the human body. There's one called nebulin that's also very big. So just because the average is 100,000 doesn't mean that every one of them is 100,000. Some of them are like dystrophin that are 2 million, 2 megabases. And then we saw insulin, which is really small, just 6,000 or so bases. So I want you to think about that, because some of these cells will express some of these genes and some of them will turn them off. And that's what makes them different. That's why a red blood cell is expressing hemoglobin. But a lot of the other cells are not going to express hemoglobin. They don't need hemoglobin. That's not their job. Their job is to be part of the eye or part of the liver or part of whatever. Each cell will express those different genes. And that's it is kind of crazy if you think about it, though, that every single cell technically has all the genes it needs to be any cell. And maybe that'll come up later as something that's going to be an important theme. And would you structure, if you had to design your own organism, is that the way you would want it? That every single unit had the ability to be any other unit in the whole machine, but that each unit would just turn on the parts that it needed? A very interesting way to look at it. I don't know that you'd necessarily think about designing a machine that way. Maybe you would. Maybe we should learn from the body how we should design machines. All right. Different cells express different genes. That is, they use their genes to produce some RNA and proteins and not others, depending on their internal state. There's like a computer word. And on cues that they and their ancestors have received from their surroundings, mainly signals from other cells in the organism. The DNA, therefore, is not just a shopping list specifying the molecules that every cell must make, and a cell is not an assembly of all the items on the list. Each cell is capable of carrying out a variety of biological tasks, depending on its environment and its history, and it selectively uses the information encoded in its DNA to guide its activities. Later in the book, we'll see in detail how DNA defines both the parts list of the cell and the rules that decide when and where these parts are to be made. And that's the interesting thing here, is that you think about DNA as this very dry list of, okay, here's a gene, here's a gene, here's a gene. There's 20,000 of these entries. And that, well, those entries, when processed, will inevitably through, obviously RNA in the middle, will become these proteins. And that's another list. But how where in this list does it say, this is something that's easy to understand, right? But where in this list does it say, okay, I cell, be an eye? Right? Where does it say in this list, lung cell, be a lung? This is a very poor drawing of a lung. 2 lungs. Where does it say kidney cell, be a kidney? And hundreds of cells, cell types that can create these organs. Where does it say, you do this, you do that, you become this, you become that? It's actually very detailed and difficult to explain. There's no really easy way to do that. Of course, it's got to be in here somewhere, right? But it's a little confusing because if each of these genes codes for a protein, those proteins are the machines that make us up, well, which part of this is sort of doing that meta or epi instruction? These are 2 important prefixes that we're going to see over and over again. Meta and epi. Where are those like instructions for the instructions? Instructions on how to use these instructions. Where do they live? That's one of the most interesting questions. Cells under the microscope. All right. Today we have access to many powerful technologies for deciphering the principles that govern the structure and activity of the cell. But cell biology started without these modern tools. Think about the earliest cell biologists began by simply looking at tissues and cells and later breaking them open or slicing them up, attempting to view their contents. What they saw was to them profoundly baffling, a collection of tiny objects whose relationship to the properties of living matter seemed an impenetrable mystery, the same way that probably protons, electrons, and neutrons seem very abstract relative to the way we think about classical physics. Nevertheless, this type of visual investigation was the first step towards understanding tissues and cells, and it remains essential today in the study of cell biology. Cells were not made visible until the 17th century when the microscope was invented. For hundreds of years afterwards, all that was known about cells was discovered using this instrument. Light microscopes use visible light to illuminate specimens, and they allowed biologists to see for the first time the intricate structure that underpins all living things. Although these instruments now incorporate many sophisticated improvements, the properties of light, specifically its wavelength, limit the fineness of detail these microscopes reveal. Does anybody know what that's called? Like the optical diffraction limit or something like that, right? There's a limit that you just can't bend the light any more than you can after a certain point. I forget what it's called exactly. It's got a name. It's not the uncertainty principle. It's called something else. It's not the Planck constant, it's the light. Yeah, I think it's just called the diffraction limit or something like that. Optical diffraction limit. Breaking the resolution limit in light microscopy. I think microscopy is one of the coolest things to study, but it's very complex, and it's all about optics, which is physical science, not chemistry or biology. But the two definitely this is where they meet in many ways. I'll read this later. Seems cool. All right, let's keep reading here. Electron microscopes, invented in the 1930s, go beyond this limit by using beams of electrons instead of beams of light as the source of illumination. Because electrons have a much shorter wavelength, that makes sense, these instruments greatly extend our ability to see the fine details of cells and even render some of the larger molecules visible individually. In this section, we describe various forms of light and electron microscopy. These vital tools in this modern cell biology laboratory continue to improve, revealing new and sometimes surprising details about how cells are built and how they operate. The invention of the light microscope led to the discovery of cells. By the 17th century, glass lenses were powerful enough to permit the detection of structures invisible to the naked eye. Using an experiment or an instrument equipped with such a lens, Robert Hooke examined a piece of cork, and in 1665, reported to the Royal Society of London that the cork was composed of a mass of minute chambers. He called these chambers cells, based on their resemblance to the simple rooms occupied by monks in a monastery. The name stuck, even though the structures Hooke described were actually the cell walls that remained after the plant cells living inside them had died. Later, Hooke and his Dutch contemporary Antony van Leeuwenhoek were able to observe living cells, seeing for the first time a world teeming with motile microscopic organisms. Can you imagine being the first person to ever see cells microscopically? What a joy that would be, that you were the first person that was able to actually see a living cell. It's like one of the defining moments in human history. What a lucky group there. For almost 200 years, such instruments, the first light microscopes, remained exotic devices, available only to a few wealthy individuals. It was not until the 19th century that microscopes began to be widely used to look at cells. The emergence of cell biology as this distinct science was a gradual process to which many individuals contributed. But its official birth is generally said to have been signaled by two publications, one by the botanist Matthias Schleiden in 1838, and the other by the zoologist Theodor Schwann in 1839. Schwann was actually a very important person. In these papers, Schleiden and Schwann documented the results of a systematic investigation of plant and animal tissues with the light microscope, showing that cells were the universal building blocks of all living tissues. So this is not too far in the distant past. This is 200 years ago, maybe somebody's great-grandfather. Not that far ago. Less than 200 years ago. Their work and that of other 19th century microscopists slowly led to the realization that all living cells are formed by the growth and division of existing cells, a principle sometimes referred to as the cell theory. The implication that living organisms do not arise spontaneously, but can be generated only from existing organisms was hotly contested, but it was finally confirmed. We'll talk about that in a second. Figure 1.5, we're talking about new cells formed by growth and division of existing cells. In 1880, Edward Strasburger drew a living plant cell, a hair cell from Tradescantia flower, which he observed dividing in two over a period of 2.5 hours. Inside the cell, the DNA, which is black, can be seen condensing into chromosomes, which are then segregated into the daughter cells. This is amazing that this is from 1880, about 140 years ago. A comparable living plant cell photographed through a modern microscope. We're going to look at that in a second and see how they differed. Almost the same, right? This is an actual photograph, and this is at a 50 micrometer resolution, so well below the limit of the naked eye. All right, so this cell theory is finally confirmed in the 1860s by an elegant set of experiments performed by Louis Pasteur. And Pasteur is really, really important in the history of biology. The principle that cells are generated only from pre-existing cells and inherit their characteristics from them underlies all of biology and gives the subject a unique flavor. In biology, questions about the present are inescapably linked to conditions in the past. To understand why present-day cells and organisms behave as they do, we need to understand their history all the way back to the misty origins of the first cells on Earth. Darwin provided the key insight that makes this history comprehensible. His theory of evolution, published in 1859, so we're getting a little closer to modern times, explains how random variation and natural selection give rise to diversity among organisms that share a common ancestry. When combined with the cell theory, the theory of evolution leads us to view all of life, from its beginnings to the present day, as one vast family tree of individual cells. Although this book is primarily about how cells work today, we'll encounter the theme of evolution again and again. light microscopes reveal some of the cell's compartments. If a very thin slice is cut from suitable plant or animal tissue and viewed under a light microscope, it is immediately apparent that the tissue is divided into thousands of small cells. In some cases, the cells are closely packed. In others, they are separated from one another by an extracellular matrix, or ECM. This is important. This is a dense material often made of protein fibers embedded in a gel of long sugar chains. ECM. I want you to remember the extracellular matrix. It's kind of what is in between cells. Each cell is typically 5 to 20 micrometers in diameter. 5 to 20 micrometers in diameter. We talked about that earlier, but I want you to always remember the size of these things and the weight. And we're going to talk about weight at some point. But these numbers matter a lot. You're not going to see a lot of numbers in biology, but I think that it's important when you do encounter numbers that you remember them. It's very embarrassing when you ask somebody how big a cell is and they're not sure. So understanding the scope of some of these things, like how big is a methane molecule and how big is a cell, it's important. The same way you could tell me how big a baseball is relative to how big a baseball stadium is. I want you to be able to do the same thing with things that are really small. All right, if care has been taken to keep the specimen alive, particles will be seen moving around inside its individual cells. On occasion, a cell may even be slowly changing shape and dividing into two. Distinguishing the internal structure of a cell is difficult, not only because the parts are small, but also because they are transparent. This is the most important concept here in microscopy. And mostly colorless, right? One way around the problem is to stain the cells with dyes that color particular components differently. Alternatively, one can exploit the fact that the cell components differ slightly from one another in refractive index, just as glass differs in refractive index from water, causing light rays to be reflected as they pass from one medium into another. Let's take a break to read question 1.3. You've embarked on an ambitious research project to create life in a test tube. You boil up a rich mixture of yeast extract and amino acids in a flask, along with a sprinkling of inorganic salts known to be essential for life. You seal the flask and allow it to cool. After several months, the liquid is as clear as ever, and there are no signs of life. A friend suggests that excluding the air was a mistake, since most life we know requires oxygen. You repeat the experiment, but this time you leave the flask open to the atmosphere. To your great delight, the liquid becomes cloudy after a few days, and under a microscope you see beautiful small cells that are clearly growing and dividing. Does this experiment prove that you managed to generate a novel life form? How might you design your experiment to allow air into the flask, yet eliminate the possibility that contamination by airborne microorganisms is the explanation for the results? There's a classic experiment by Louis Pasteur that shows the answer. But of course, this experimenter would have made the error that allowing that oxygen in would also allow microorganisms in, so they did not create life. They would have to pump in pure oxygen from a vacuum or something like that. Let's look at figure 1.6. Cells form tissues in plants and animals. This is panel A. Cells in the root tip of a fern. The DNA containing nuclei are stained red, and each cell is surrounded by a thin cell wall, light blue. The red nuclei of densely packed cells are seen at the bottom corners of the preparation. This is cell panel B. Cells in the crypts of the small intestines. Yes, they're called crypts. Isn't that crazy? Crypt cells. Each crypt appears in the cross-section as a ring of closely packed cells with nuclei stained blue. The ring is surrounded by extracellular matrix, the ECM I mentioned earlier, which contains the scattered cells that produce most of the matrix components. All right, so light rays deflecting from one to another. The small differences in refractive index can be made visible by specialized optical techniques, and the resulting images can be enhanced further by electronic processing. As shown in figures 1.6b and 1.7a, typical animal cells visualized in these ways have a distinct anatomy. They have a sharply defined boundary, indicating the presence of an enclosing membrane called the plasma membrane, sometimes also called the cell membrane. But plasma membrane is probably the better phrase. The large, round structure called the nucleus. This is also a term we use in physics and chemistry, not to be confused though. The nucleus is prominent near the middle of the cell. Around the nucleus and filling the cell's interior is the cytoplasm. These are all really important words. I haven't bolded a lot of stuff in this book, but the stuff I'm bolding now is really critical. The cytoplasm is a transparent substance crammed with what seems at first to be a jumble of miscellaneous objects. With a good light microscope, one can begin to distinguish and classify some of the components in the cytoplasm, but structures smaller than about 0.2 micrometer, about half the wavelength of visible light, cannot normally be resolved. Points closer than this are not distinguishable and appear as a single blur. So that's kind of the diffraction limit I was talking about. If you know your wavelengths, you know that red is what, around 400 nanometers, which is around, or exactly, 0.4 micrometers, right? And then going all the way to blue, or violet, it's around 800 nanometers. So when people say, what's your favorite color, I say around 600. But the point is that by using light, that's the best we can do with the wavelength to use diffraction. So we have to use something less than 400 nanometers. And eventually, when you get small enough, you can use something really, really small. In recent years, however, new types of light microscope called fluorescence microscopes have been developed that use sophisticated methods of illumination and electronic image processing to see fluorescently labeled cell components in much finer detail. The most recent super-resolution fluorescent microscopes, for example, can push the limits of resolution down even further to about 20 nanometers. That's the size of a single ribosome, which is a protein, a large macromolecular complex in which RNAs are translated into proteins. So the ribosome translates RNA into proteins. Interesting. That sounds really important. Ribosome. So 20 nanometers is not quite the size of an atom, right? But it's the size of sort of a protein. So we think about like micrometer seems to be cell scale. Nanometer seems to be protein scale. And maybe this is sort of the high nanometers, and low nanometers / picometers is the atom scale. And we don't really go below the atom scale, right? That's the smallest scale that we care about. Then maybe the millimeters is the organ scale. Organs are made up of cells, so that's one way to look at it as well. Organs and tissues. So these are sort of different scales that we can think about in biology. All right, let's look at these techniques in 1.7. Some of the internal structures of a cell can be seen with a light microscope. A cell taken from human skin and grown in culture was photographed through a light microscope using interference contrast optics. This is A. We see a nucleus, we have the plasma membrane, and the cytoplasm. This is a 40 micrometer scale, so the entire width is maybe, 200 micrometers. A pigment cell. This is a pigment cell from a frog. Stained with fluorescent dyes and viewed with a confocal fluorescent microscope. The nucleus is in purple, the pigment granules are in red. Cool. And the microtubules, a class of protein filaments in the cytoplasm, is green. Very cool. All right, the fine structure of a cell is revealed by electron microscopy. For the highest magnification and best resolution, one must turn to an electron microscope, which can reveal details down to a few nanometers. I have one in the back. Preparing cell samples for the electron microscope is a painstaking process. Even for light microscopy, a tissue often has to be fixed, that is, preserved by pickling in a reactive chemical solution, supported by embedding in a solid wax or resin, cut or sectioned into thin slices and stained before its view. The tissues in figure 1.6 were prepared in this way. For electron microscopy, similar procedures are required, but the sections have to be much thinner, and there is no possibility of looking at living cells. When thin sections are cut, stained with electron-dense heavy metals, and placed in an electron microscope, much of the jumble of cell components becomes sharply resolved into distinct organelles, separate recognizable substructures with specialized functions that are often hazily defined with a conventional light microscope. A delicate membrane, only about 5 nanometers thick, is visible enclosing the cell, and similar membranes form the boundary of many organelles inside. The plasma membrane separates the interior of the cell from its external environment, while internal membranes surround most organelles. All these membranes are only 2 molecules thick. 2 molecules thick. With an electron microscope, even individual large molecules can be seen. So we're going to look at the fine structure of a cell can be seen in a transmission electron microscope, or a TEM, T-E-M. A, the thin section of a liver cell, showing the enormous amount of detail that is visible. Some of the components to be discussed later in the chapter are labeled. They are identifiable by their size, location, and shape. So this is a peroxisome, this is a lysosome, this is a mitochondria, and this is the endoplasmic reticulum, or the ER. There's the plasma membrane, and there's the nucleus. Let's see what's in cell B. A small region of the cytoplasm at a higher magnification. The small structures that are clearly visible are the ribosomes, each of which is made of 80 or so individual protein and RNA molecules. Some of the ribosomes are free in the cytoplasm, while others are bound to a membrane-enclosed organelle, the ER, endoplasmic reticulum, discussed later. I'd say ER is a really important concept. So this is in panel B. We have the ribosomes, the endoplasmic reticulum, and the mitochondrium. Mitochondria, plural. And this is at 2 micrometers, so this is really zoomed in. This is 2 micrometers, but not as zoomed in. So here's 50 nanometers, so this is extreme zoom, and we can see a DNA molecule. And again, it's very zoomed out. It looks like it could be the surface of Mars or something, right? Portion of a long thread-like DNA molecule isolated from a cell and viewed by electron microscopy. So that's what panel C was. All right. The type of electron microscope used to look at thin sections of tissues is known as a transmission electron microscope, a TEM. This instrument is, in principle, similar to a light microscope, except that it transmits a beam of electrons rather than a beam of light through a sample. Another type of electron microscope is a scanning electron microscope, SEM, which scatters electrons off the surface of a sample and so is used to look at the surface details of cells and other structures. These techniques, along with different types of light microscopy, are reviewed in panel 1.1. Even the most powerful electron microscopes, however, cannot visualize the individual atoms that make up biological molecules. To study the cell's key components in atomic detail, biologists have developed even more sophisticated tools. Techniques such as X-ray crystallography or cryo-electron microscopy, or cryo-EM, important idea, relatively new, for example, can be used to determine the precise positioning of atoms within three-dimensional structure of proteins and their complexes, which we'll discuss in chapter 4. How big are cells and their components? This chart lists sizes of cells and their component parts, the units in which they're measured, and the instruments needed to visualize them. These drawings convey a sense of scale between living cells and atoms. Each panel shows an image that is magnified by a factor of 10 compared to its predecessor, producing an imaginary progression from a thumb to skin cells to a mitochondrion to a ribosome to ultimately a cluster of atoms that forming part of one of the many protein molecules in our bodies. Note the ribosomes are present inside mitochondria as well as in the cytoplasm. Details of molecular structure shown in the last 2 bottom panels are beyond the power of the electron microscope. So let's start with our thumb. This is somebody's thumb, and we take this little piece and magnify it from 20 millimeters. This is very visible, right? 20 millimeters. All the way to 2 millimeters, which is less visible, but we can see some detail there. And then finally to 0.2 millimeters, which is, 200 micrometers. And now we're looking at, we can see individual cells, which we couldn't see before. Now at 20 micrometers, we can not only see individual cells, we can see some cell components. At 2 micrometers, we can actually see an entire cell component. and we can see the mitochondria, for example. We see ribosomes, but not in good detail. At 0.2 micrometers, or 200 nanometers, we can now see the ribosomes. At 20 nanometers, very, very small indeed, we can see the actual ribosomes and even little parts of the protein. So these are proteins. And at 2 nanometers, we see the proteins. At 0.2 nanometers, these are individual atoms. This is the angstrom length. So we can see the scale here, and I would highly recommend you study this and think about it closely. Let's move on to a new section, the prokaryotic cell. Of all the types of cells that have been examined microscopically, bacteria have the simplest structure and come closest to showing us life stripped down to its bare essentials. There's this interesting experiment where Craig Venter, who was the first person to sequence the human genome, is also trying to make a new OS of the operating system for a new synthetic cell, which would be like designed from scratch. And this is kind of what bacteria is in the first place, but it'd be interesting to see if one could create it from scratch by oneself. Anyway, a bacterium contains no organelles other than the ribosomes, not even a nucleus to hold its DNA. So these bacteria don't have nuclei. Interesting, right? This property, the presence or absence of a nucleus, is used as the basis for a simple but fundamental classification of all living things. Organisms whose cells have a nucleus are called eukaryotes. Eu means well in Greek, or truly. Karyon means nucleus. So you're going to see karyotyping, karyo comes up a lot. Eu comes up in a lot of different sciences and other words like euphemism or something, right? Organisms whose cells do not have a nucleus are called prokaryotes. Prokaryotes. Prokaryotes are typically spherical, rod-like, or corkscrew-shaped. They're also small, generally just a few micrometers long, although some giant species are as much as 100 times longer than this. Prokaryotes often have a tough protective coat or cell wall surrounding the plasma membrane, so they're like two cell walls, which encloses a single compartment which contains the cytoplasm and DNA, kind of all mixed up in one soup. In the electron microscope, the cell interior typically appears as a matrix of varying texture without any obvious organized internal structure. The cells reproduce quickly by dividing into two. Under optimum conditions, when food is plentiful, many prokaryotic cells can duplicate themselves in as little as 20 minutes. In only 11 hours, a single prokaryote can therefore give rise to more than 8 billion progeny. 8 billion from one. Amazing. That's the power of exponents, I guess. Thanks to their large numbers, rapid proliferation, and ability to exchange bits of genetic material by a process akin to sex, they're sort of autosexual. Populations of prokaryotic cells can evolve fast, rapidly acquiring the ability to use a new food source or resist being killed by a new antibiotic. Interesting. Let's look at these pages we skipped. This is panel 1.1, talking about microscopy. In conventional light microscopy, we have our retina. This is our eye, an eyepiece. The tube lens, an objective lens. Here's the specimen that we prepared on a glass slide. There's a condenser lens and a light source. The light path in this light microscope, hits our retina. A conventional light microscope allows us to magnify cells up to 1,000 times and resolve details as small as 200 nanometers, a limitation imposed by the wave-like nature of light, not by the quality of the lenses. 3 things are required for viewing cells in a light microscope. First, a bright light must be focused onto the specimen by lenses in the condenser. Second, the specimen must be carefully prepared to allow light to pass through it. Third, an appropriate set of lenses, an objective tube and eyepiece, must be arranged to focus an image of the specimen. Looking at live cells, the same unstained living animal cell, a fibroblast, maybe we should start here, or maybe not. So this is a fibroblast, is viewed with A, the simplest bright field optics, B, phase contrast optics, and C, interference contrast optics. You can see the quite different qualities. The 2 latter systems exploit differences in the way light travels through regions of the cell with differing refractive indices. All 3 images can be obtained on the same microscope simply by interchanging optical components.
fixed samples. Most tissues are neither small enough nor transparent enough to examine directly in the microscope. Therefore, they're chemically fixed and cut into thin slices or sections that can be mounted on a glass microscope slide and subsequently stained to reveal different components of the cells. A stained section of a plant root tip is shown here. All right, let's look at this fluorescence microscopy. So we have an eyepiece, we have a light source, and then we have a beam splitting mirror. And then there's an objective lens here, and of course the object, or specimen. Fluorescent dyes used for staining cells are detected with the aid of a fluorescence microscope. This is similar to an ordinary light microscope, except that the illuminating light is passed through two sets of filters. The first filters the light before it reaches the specimen, passing only those wavelengths that excite the dye. The second blocks out this light and passes only the wavelengths emitted when the dye fluoresces. Dyed objects show up in bright color on a dark background. Fluorescent probes. Fluorescent molecules absorb light at one wavelength and emit it at another longer wavelength. Some fluorescent dyes bind specifically to particular molecules in cells and can reveal their location when the cells are examined with a fluorescence microscope. In these dividing nuclei, in a fly embryo, the stain for DNA fluoresces blue. Other dyes can be coupled to antibody molecules, which then can serve as highly specific staining reagents that bind selectively to particular molecules, showing their distribution in the cell. Because fluorescent dyes emit light, they allow objects even smaller than 0.2 micrometers to be seen. Here, a microtubule protein in the mitotic spindle is stained green with a fluorescence antibody. So think about that. Fluorescent dyes emit light, so they allow objects even smaller than 2 micrometers to be seen. Why is that true? It's a good question. Confocal fluorescence microscopy. A confocal microscope is a specialized type of fluorescence microscope that builds up an image by scanning the specimen with a laser beam. The beam is focused onto a single point of specific depth of the specimen, and a pinhole aperture in the detector allows only fluorescence emitted from the same point to be included in the image. Scanning the beam across the specimen generates a sharp image of the plane of focus, an optical section. The series of optical sections at different depths allow a 3D image to be constructed, such as this highly branched mitochondrion in a living yeast cell. Pretty cool. Super resolution fluorescence microscopy. Several recent and ingenious techniques have allowed fluorescence microscopes to break the usual resolution limit of 200 nanometers. One such technique uses a sample that is labeled with molecules whose fluorescence can be reversibly switched on and off by different colored lasers. The specimen is scanned by a nested set of 2 laser beams, in which the central beam excites fluorescence in a very small spot of the sample, while a second beam, wrapped around the first, switches off fluorescence in the surrounding area. A related approach allows the positions of individual fluorescent molecules to be accurately mapped while others nearby are switched off. Both approaches slowly build up an image with a resolution as low as 20 nanometers. These new super resolution methods are being extended into 3D imaging and real-time live cell imaging. These are microtubules viewed with a conventional fluorescence microscope on the left and with a super resolution optics on the right. In the super resolution image, the microtubule can be clearly seen at the actual size, which is only 25 nanometers in diameter. All right, let's look at the TEM, pretty big instrument here. The electron micrograph below shows a small region of a cell in a thin section of testes. The tissue has been chemically fixed, embedded in plastic, and cut into very thin sections that have been stained with salts of uranium and lead. That's the usual staining for a TEM. The TEM is in principle similar to a light microscope, but it uses a beam of electrons whose wavelength is very short instead of a beam of light, and magnetic coils to focus the beam instead of glass lenses. Because of the very small wavelength of electrons, the specimen must be very thin. This is 500 nanometers resolution. Contrast is usually introduced by staining the specimen with electron-dense heavy metals. Lead has a lot of electrons. The specimen is then placed in a vacuum in the microscope. The TEM is a useful magnification of up to 1,000,000-fold and can resolve details as small as about 1 nanometer, which is a billionth of a meter in biological specimens. So we have an electron gun, condenser lens, the specimen, objective lens, projector lens, and viewing screen or photographic film. This is a SEM, scanning electron microscopy. Similar concept, except there's a beam deflector here, and electrons from the specimen bounce off the specimen, and there's a detector that interprets it. In a scanning electron microscope, the specimen, which has been coated with a very thin film of a heavy metal, is scanned by a beam of electrons brought to a focus on the specimen by magnetic coils that act as lenses. The quantity of electrons scattered or emitted as the beam bombards each successive point on the surface of the specimen is measured by the detector and is used to control the intensity of successive points in an image built up on a video screen. The microscope creates striking images of three-dimensional objects with great depth of focus and can resolve details down to 3 to 20 nanometers, depending on the instrument. So you can see here a pretty beautiful SEM image called a micrograph of stereocilia projecting from a hair cell in the inner ear. For comparison, the same structure is shown by light microscopy at the limit of resolution. Not very clear there. Very cool stuff. All right, in this section we offer an overview of the world of prokaryotes. Despite their simple appearance, these organisms lead sophisticated lives, occupying a stunning variety of ecological niches. We will also introduce the two distinct classes into which prokaryotes are divided, bacteria and archaea. Singular is archaeon. Although they are structurally indistinguishable, archaea and bacteria are only distantly related. Interesting. Bacteria come in different shapes and sizes. Typical spherical, rod-like, and spiral-shaped bacteria are shown and are drawn to scale. The spiral cells shown are the organisms that cause syphilis. Here they are. Treponema pallidum, syphilis bacteria. Rod-shaped cells like E. coli and Salmonella. And spherical cells like Streptococcus. 2 micrometers is the resolution here. All right, question 1.4. A bacterium weighs about 10 to the negative 12th grams. So what's that? A picogram. And can divide every 20 minutes. If a single bacterial cell carried on dividing at this rate, how long would it take before the mass of the bacteria would equal that to the Earth? The Earth weighs 6 times 10 to the 24th kilograms. Contrast your result with the fact that bacteria originated at least 3.5 billion years ago and have been dividing ever since. Explain the apparent paradox. The number of cells, n, in a culture at a time t, is described by the equation n equals n sub 0 times 2 to the t over g, where n sub 0 is the number of cells at 0 time and g is the population doubling time. So basically you would expect bacteria to overrun the whole planet over time, right? But that is not what's happened. And I think we have an obvious answer there, right? The bacteria don't only divide, they also die. Prokaryotes are the most diverse and numerous cells on Earth. Most prokaryotes live as single-celled organisms. Although some join together to form chains, clusters, or other organized multicellular structures. In shape and structure, prokaryotes may seem simple and limited, but in terms of chemistry, they're the most diverse class of cells on the planet. Members of this class exploit an enormous range of habitats, from hot puddles of volcanic mud to the interiors of other living cells, and they vastly outnumber all eukaryotic organisms on Earth. Some are aerobic, using oxygen to oxidize food molecules, very important. And some are strictly anaerobic and are killed by the slightest exposure to oxygen. As we discuss later in this chapter, mitochondria, the organelles that generate energy in eukaryotic cells, are thought to have evolved from aerobic bacteria. Which is a really crazy concept. Let's look at figure 1.11 really quickly. The bacterium E. coli has served as an important model organism. This is an electron micrograph of a longitudinal section of E. coli. The cell's DNA is concentrated in the lightly stained region. Notice there's no nucleus. E. coli is an outer membrane and an inner plasma membrane, so he's got 2 cell membranes and a thin wall in between. The many flagella distributed over its surface are not visible in the micrograph. So this is a whole cell is maybe 2 micrometers of diameter. So anyway, mitochondria, this is one of the coolest things I learned. I actually did a special report and course on this concept, that mitochondria evolved from bacteria. So our mitochondria in our cells now live inside of the anaerobic ancestors of today's eukaryotic cells. Thus, our own oxygen-based metabolism can be regarded as the product of the activities of bacterial cells. So it's kind of a symbiosis. Virtually any organic carbon-containing material from wood or petroleum can be used as food by one sort of bacterium or another. Even more remarkably, some prokaryotes can live entirely on inorganic substances. They can get their carbon from CO2 in the atmosphere, their nitrogen from N2 in the atmosphere, or air, and their oxygen, hydrogen, and sulfur and phosphorus from air, water, and inorganic materials. Some of these prokaryotic cells, like plant cells, perform photosynthesis. They use energy from the sunlight to produce organic molecules from carbon dioxide. Others derive energy from the chemical reactivity of inorganic substances in the environment. In either case, such prokaryotes play a unique and fundamental part of the economy of life on Earth, as other living organisms depend on the organic compounds that these cells generate from inorganic materials. So there's a whole life cycle that occurs. What else did I want to mention? Yeah, just this idea that, again, our mitochondrion probably came from bacteria. And if you look at the way mitochondria are structured, they kind of look like their own little cells within our cells, and they even have their own DNA, which is really crazy. Let's look at figure 112 here. Some bacteria are photosynthetic. Anabaena cylindrica forms long multicellular chains. This light micrograph shows specialized cells that either fix nitrogen, that is, they capture nitrogen from the atmosphere and incorporate it into organic compounds. We'll label that H. Or they fix CO2 from photosynthesis, label that V, or they become resistant spores, labeled S, that can survive under unfavorable conditions. Panel B on the bottom is an electron micrograph of a related species called Formidium laminosum, which shows the intracellular membranes where photosynthesis occurs. As shown in these micrographs, some prokaryotes can have intracellular membranes and form simple multicellular organisms. If you're just joining us, we're going through a Harvard curriculum. The first class is biology, cell biology, and we're wrapping up chapter 1. Then we're going to go to a different textbook and do class 2 of a curriculum of your average Harvard undergraduate student, at least maybe first year. Plants, too, can capture energy from sunlight and carbon from atmospheric CO2. The plants unaided by bacteria cannot capture nitrogen from the atmosphere. In a sense, plants even depend on bacteria for photosynthesis. As we discussed later, it is almost certain that the organelles in the plant cell that perform photosynthesis, the chloroplasts, have evolved from photosynthetic bacteria that long ago found a home inside the cytoplasm of a plant cell ancestor. So you'll see this concept of symbiosis occur repeatedly. The world of prokaryotes is divided into 2 domains, bacteria and archaea. Traditionally, all prokaryotes have been classified together in one large group, but molecular studies have determined that there is a gulf within the class of prokaryotes, dividing into 2 distinct domains, the bacteria and the archaea, which are thought to have diverged from a common prokaryotic ancestor about 3.5 billion years ago. Remarkably, DNA sequencing reveals that at the molecular level, the members of these 2 domains differ as much from one another as either does from the eukaryotes. Interesting. Most of the prokaryotes familiar from everyday life, the species that live in the soil or make us ill, are bacteria. Archaea are found not only in these habitats, but also in environments that are too hostile for most other cells. Brine, hot acid of volcanic springs, airless depths of marine sediments, the sludge of sewage treatment plants, pools beneath the frozen surface of Antarctica, as well as in the acidic, oxygen-free environment of a cow's stomach, where they break down ingested cellulose and generate methane gas. Many of these extreme environments resemble the harsh conditions that must have existed on the primitive Earth, where living things first evolved before the atmosphere became rich in oxygen. Here is a sulfur bacterium, and it gets its energy from H2S. This is called chemosynthesis. Vegetoa, a prokaryote that lives in sulfurous environments, can oxidize H2S to produce sulfur and can fix carbon even in the dark. In this light micrograph, yellow deposits of sulfur can be seen inside 2 of the bacterial cells. Isn't that amazing? All right, the eukaryotic cell. Eukaryotic cells in general are bigger and more elaborate than bacteria and archaea. Some live independent lives as single-cell organisms, such as amoeba and yeasts. Others live in multicellular assemblies. All of the more complex multicellular organisms, including plants, animals, and fungi, are formed from eukaryotic cells. So plants, animals, and fungi. Eukaryotic. Don't forget. Figure 114. Yeasts are simple, free-living eukaryotes. The cells shown in this micrograph belong to a species of yeast, Saccharomyces cerevisiae, used to make dough rise and turn malted barley juice into beer. That's where you get cerevisiae, right? As seen in this image, the cells reproduce by growing a bud and then dividing asymmetrically into a mother cell and a small daughter cell. For this reason, they are called budding yeast. All right, by definition, all eukaryotic cells have a nucleus. That's why they're eukaryotes. But possession of a nucleus goes hand in hand with possession of a variety of other organelles, most of which are membrane enclosed and common to all eukaryotic organisms. In this section, we'll take a look at the main organelles found in eukaryotic cells from the point of view of their functions, and we consider how they came to serve the roles they have in the life of the eukaryotic cell. The nucleus is the information store of the cell, extremely important. The nucleus is usually the most prominent organelle in a eukaryotic cell. Right? The nucleus contains most of the DNA in the eukaryotic cell. This drawing of a typical animal cell shows its extensive system of membrane-enclosed organelles. The nucleus is colored brown, and the nuclear envelope is green. And the cytoplasm, the interior cell outside of the nucleus, is white. Panel B here is an electron micrograph of the nucleus in a mammalian cell. Individual chromosomes are not visible because at this stage of the DNA cycle, or the cell division cycle, the DNA molecules are dispersed as fine threads around the nucleus. Here are the mitochondria, which we'll also talk about. All right, so what is the nucleus? It is enclosed within two concentric membranes, okay, that form the nuclear envelope. And it contains molecules of DNA, extremely long polymers that encode the genetic information of the organism. We talked about DNA a minute ago. In the light microscope, these giant DNA molecules become visible as individual chromosomes. Right? We have 23 pairs of chromosomes. When they become more compact before a cell divides into two daughter cells. DNA also carries the genetic information in prokaryotic cells, but these cells lack a distinct nucleus not because they lack DNA, but because they don't keep their DNA inside a nuclear envelope, which is segregated from the rest of the cell contents. Here's the nucleus, here's the nuclear envelope, and here is before division, and here, as we get closer to cell division, we can see that the chromosomes will start to condense and form. So figure 116, chromosomes become visible when a cell is about to divide. As the eukaryotic cell prepares to divide, its DNA molecules become progressively more compacted or condensed, forming worm-like chromosomes that can be distinguished in the light microscope. The photographs shown here show 3 successive steps in the chromosome condensation process in a cultured cell from a newt's lung. Note that the last micrograph on the right, the nuclear envelope has broken down. Mitochondria. We always joke about mitochondria being the powerhouse of the cell, right? But there's more to it than that. Mitochondria generate usable energy from food. Mitochondria are present in all eukaryotic cells, and they are among the most conspicuous organelles in the cytoplasm. In a fluorescence microscope, they appear as worm-shaped structures that often form branching networks. When seen with an electron microscope, individual mitochondria are found to be enclosed in 2 separate membranes, with the inner membrane formed into folds that project into the interior of the organelle. Microscopic examination by itself, however, gives little indication of what the mitochondria do. Their function was discovered by breaking open cells and then spinning the soup of cell fragments in a centrifuge. This treatment separates the organelles according to their size and density. That's what a centrifuge does. Purified mitochondria were then tested to see what chemical processes they could perform. This revealed the mitochondria are generators of chemical energy for the cell. They harness the energy from the oxidation of food molecules, such as sugars, to produce adenosine triphosphate, or ATP, probably the most important molecule in this book. The basic chemical fuel that powers most of the cell's activities. Because the mitochondria consumes oxygen and releases CO2 in the course of this activity, this entire process is called cell respiration. Essentially breathing at the level of the cell. Take in oxygen, release CO2. Without mitochondria, animals, fungi, and plants would be unable to use oxygen to extract the energy they need from the food molecules that nourish them. The process of cell respiration is in chapter 14. Mitochondria contain their own DNA. This blew my mind. And this kind of gives you the evidence that they were originally their own organisms and that we basically merged with them. Isn't that crazy? And they reproduce by dividing. Because they resemble bacteria in so many ways, they're thought to derive from bacteria that were engulfed by some ancestor of present-day eukaryotes. Mitochondria vary in shape and size. This budding yeast cell, which contains a green fluorescent protein in its mitochondria, was viewed in a super-resolution confocal fluorescence microscope. In this 3D image, the mitochondria are seen to form complex branch networks. Look pretty different from what you normally see mitochondria as. So anyway, some ancestor engulfed this bacteria that we now call mitochondria. This evidently created a symbiotic relationship in which the host eukaryote and the engulfed bacterium helped each other to survive and reproduce. Makes sense, right? Mitochondria have a distinct internal structure. We're going to look at an electron micrograph of a cross-section of mitochondria revealing extensive infolding of the inner membrane. It's very much like a maze, right? There's an artist's drawing. There's an outer membrane and inner membrane. Three-dimensional representation of this arrangement of the mitochondrial membrane shows the smooth outer membrane and the highly convoluted inner membrane. The inner membrane contains most of the proteins responsible for energy production. It is highly folded to provide a large surface area. That's usually why things are folded in the body for this activity. In this schematic cell, the innermost compartment of the mitochondria is colored orange. There it is. Mitochondria are thought to have evolved from engulfed bacteria. It is virtually certain that mitochondria evolved from aerobic bacteria that were engulfed by an archaea-derived early anaerobic eukaryotic cell and survived inside of it, living in symbiosis with their host. As shown in this model, the double membrane of the present-day mitochondria is thought to have been derived from the plasma membrane and outer membrane of the engulfed bacterium. The membrane derived from the plasma membrane of the engulfing ancestral cell was ultimately lost. There's the bacterium. It kind of invaded the host cell, and it just kind of lives now with us as one of the most important parts of the cell. These are early aerobic eukaryotic cells and how they kind of adopted, basically, mitochondria, which were originally bacteria. Let's talk about chloroplasts briefly. Chloroplasts capture energy from sunlight. They're large green organelles that are found in the cells of plants and algae, but not in animals or fungi. These organelles have an even more complex structure than mitochondria. In addition to their two surrounding membranes, they possess internal stacks of membranes containing the green pigment chlorophyll. You've heard of that, I'm sure. This is a chloroplast. These are chloroplasts in plant cells that capture the energy of sunlight. A single cell isolated from a leaf or flowering plant, seen in the light microscope, showing many green chloroplasts. A drawing of one of these chloroplasts showing the inner and outer membranes, as well as highly folded system of internal membranes containing the green chlorophyll molecules that absorb light energy. Chloroplasts carry out photosynthesis, trapping the energy of the sunlight in their chlorophyll molecules and using this energy to drive the manufacture of energy-rich sugar molecules. Cool. In this process, they release oxygen as a molecular byproduct. They release oxygen. That's what a plant does. Plant cells can then extract this stored chemical energy when they need it, in the same way animals do, by oxidizing these sugars and their breakdown products mainly in their mitochondria. Chloroplasts thus enable plants to get their energy directly from sunlight. They also allow plants to produce the food molecules and the oxygen that mitochondria use to generate chemical energy in the form of ATP. We'll talk about how these organelles work together in chapter 14. Like mitochondria, chloroplasts contain their own DNA, reproduce by dividing into two, and are thought to have evolved from bacteria, in this case from photosynthetic bacteria that also were engulfed by an early aerobic eukaryotic cell. All right, enough about plants. although we'll do one last thing. Chloroplasts almost certainly evolved from engulfed photosynthetic bacteria. These bacteria are thought to have been taken up by early eukaryotic cells that already contained mitochondria, so a very similar concept here. Internal membranes create intracellular compartments with different functions. I like this word intracellular. You should use it when we're talking about inside of the cell versus extracellular. This is very important. Nuclei, mitochondria, and chloroplasts are not the only membrane-enclosed organelles inside eukaryotic cells. Okay, what else is there then, Mr. Textbook? The cytoplasm contains a profusion of other organelles that are surrounded by single membranes. Most of these structures are involved with the cell's ability to import raw materials, sounds like a business, and to export both useful substances and waste products that are produced by the cell, which we'll discuss in chapter 12. Here's the first one that they mentioned, which I've called out as very important. The endoplasmic reticulum is an irregular maze of interconnected spaces enclosed by membrane. It is the site where the most cell membrane components, as well as materials destined for export from the cell are made. So it's like a factory. This organelle is enormously enlarged in cells that are specialized for their secretion of proteins. Stacks of flattened membrane-enclosed sacs constitute the Golgi apparatus, which modifies and packages molecules made in the endoplasmic reticulum that are destined to be either secreted from the cell or transported to another cell component. Lysosomes are small, irregular-shaped organelles in which intracellular digestion occurs, releasing nutrients from ingested food particles into the cytosol and breaking down unwanted molecules for either recycling within the cell or excretion from the cell. What's kind of interesting here is you have like a fractal representation. You have a cell that seems to have an input of raw materials and an output of waste. And this is at the cell level, which, as we said, is 2 to 5 micrograms, or micrometers, excuse me. But then we also have different organisms. I'm drawing my cat, who doesn't have a name yet. And the cat also basically has the same concept, right? The cat takes in internal raw materials and puts out waste. So we have this kind of like fractal relationship where at every dimension, as you zoom in, you sort of have the same concept. You could even argue that if we zoom out, all animals, not just cats, we kind of have an ecosystem that does the same thing, that takes in lots of raw materials and outputs lots of waste, not just excretion waste, but also wastes like pollution, right? So it's very interesting that you have this fractal relationship. Fractal is something that has the same nature no matter how many times you zoom in. If you want to learn more about fractals and the person that sort of discovered or invented them, Mandelbrot, I would highly encourage you to do so. All right. So we're talking about lysosomes and how they break down different molecules and digest them, basically. And those are then recycled within the cell or excreted. Many of the large and small molecules within the cell are constantly being broken down and remade. Peroxisomes are small membrane-enclosed vesicles that provide a sequestered environment for a variety of reactions in which hydrogen peroxide is used to inactivate toxic molecules. That's why they're peroxisomes. Membranes also form many types of small transport vesicles that ferry materials between one membrane-enclosed organelle and another. All these membrane-enclosed organelles are highlighted here. So we're going to go through this in a second. All right, the endoplasmic reticulum produces many of the components of the cell. This is a diagram of the animal cell that shows the ER in green. Okay, look at how important the ER is, how much volume it takes. This is an electron micrograph. This is a schematic. This is the electron micrograph of a thin section of a mammalian pancreatic cell, so from the pancreas, that shows a small part of the ER, of which there are vast amounts in this cell type, which is specialized for protein secretion. That's in fact what the pancreas does, right? It secretes hormones. Note that the endoplasmic reticulum is continuous, and the membranes, or it's continuous with the membranes of the nuclear envelope. So here's the nucleus, here's the nuclear envelope, and you can see that you have this sort of like highway, basically, between the two. The black particles studding the region of the ER, you see these little black particles, and the nuclear envelope here are shown, are actually ribosomes, which translate RNA into proteins. Because of its appearance, the ribosome-coated ER is often called the rough ER, and that's different from the smooth ER, which doesn't have ribosomes. You can see parts of it that are smooth. All right, let's look at the Golgi apparatus. This is a little bit different from the ER. The Golgi apparatus is composed of a stack of flattened membrane-enclosed discs. Take a look at this schematic A and B. This is a schematic diagram of an animal cell with Golgi in red. Here's a more realistic drawing. Flattened discs, stacked flattened discs. Some of the vesicles seen nearby have pinched off to form the Golgi stack. Others are destined to fuse with it. Only one stack is shown here, but several can be present in a cell. This is an electron micrograph that shows the Golgi apparatus from a typical animal cell, and you can see it's kind of connected to the endoplasmic reticulum. And there's the nuclear envelope. All right, membrane-enclosed organelles are distributed throughout the eukaryotic cell cytoplasm. The various types of membrane-enclosed organelles, shown in different colors, are each specialized to perform a different function. The cytoplasm that fills the space outside of these organelles is called the cytosol. This is the cytosol, which contains the cytoplasm. Nuclear envelope, nucleus, transport vesicles, ER, Golgi, peroxisomes, mitochondria, and lysosome. So now you can kind of point to a cell and see what everything does. Pretty great. All right, let's get to something really important. A continual exchange of materials takes place between the ER, the Golgi, and lysosomes, the plasma membrane, and the outside of the cell, the extracellular environment. The exchange is mediated by transport vesicles that pinch off from the membrane. I want you to think about this concept of one organelle and fuse with another, like tiny soap bubbles that bud from and combine with other bubbles. At the surface of the cell, for example, portions of the plasma membrane tuck inward. That's the pinching process here. And pinch off to form vesicles. They carry material captured from the external medium of the cell, a process called endocytosis. Animal cells can engulf very large particles or even entire foreign cells by endocytosis. In the reverse process called exocytosis, vesicles from the inside of the cell fuse with the plasma membrane and release their contents into the external medium. Most of the hormones and signal molecules that allow cells to communicate with one another are secreted from cells by exocytosis. How membrane-enclosed organelles move proteins and other molecules from place to place inside the eukaryotic cell is discussed in chapter 15. It's like a huge factory that needs like very detailed kind of instructions on how to ship everything from one place to another. All right, the cytosol is a concentrated aqueous gel of large and small molecules. This is a really good concept. If we were to strip the plasma membrane from a eukaryotic cell and remove all of its membrane-enclosed organelles, including the nucleus, ER, Golgi, mitochondria, chloroplasts, and so on, we'd be left with a cytosol. In other words, the cytosol is the part of the cytoplasm that is not contained within the intracellular membranes. In most cells, the cytosol is the single largest cell compartment. It contains a host of large and small molecules crowded together so closely that it behaves more like a gel than a liquid solution. And there's actually a really cool software program I'll show you from Scripps. It's called Mesoscale. And you can see here, it's sort of a 3D model of a cell. So there's never really been software like this. And you can actually use this on the web. Don't remember exactly where. Let's see. Maybe I'll go here. Yeah, here it is. And so if I run this, I can actually, let's look at COVID. This is going to render kind of like a cross-section of a COVID virion particle. And you can sort of see what it looks like. And so oftentimes when you do these simulations, you do them a single protein at a time, and there's not really a good tool for visualizing the entire cell. So this tool was made for that. For that purpose. Let's take a look at this tool, which is a bit more complex. It's rendering and downloading. It's very complex cell. This is a bacterial cell, or really a fungi, mycoplasma. And you can kind of see what they're talking about, that the cytoplasm is really, well, this is a bit more hollow than I would have expected, but really cool stuff. So you can play around with this software if you'd like. It's called Mesoscale or Mesoscope, and it's made by Scripps. So anyway, the cytosol is more like a water-based gel. So eukaryotic cells engage in continual endocytosis and exocytosis to traffic things across their cell membrane. They import extracellular materials by endocytosis and secrete intracellular materials by exocytosis. Endocytosed material is first delivered to membrane-enclosed organelles called endosomes, which we didn't talk about yet, but really important kind of concept there. The cytosol is extremely crowded. This is sort of what the software is looking at will make clear. The atomically detailed model of cytosol of E. coli is based on size and concentrations of 50 of its most abundant large molecules present in the bacterium. There are RNAs, proteins, and ribosomes in different colors. This is 25 nanometers, so pretty zoomed in. All right, so it's much more of a gel than a liquid. The cytosol is the site of many chemical reactions that are fundamental to the cell's existence. The early steps in the breakdown of nutrient molecules take place in the cytosol, for example, and it is here that most proteins are made by ribosomes. Okay. Can you give me a reason why it would be advantageous for eukaryotic cells to evolve elaborate internal membrane systems that allow them to import substances from the outside? I think that's fairly obvious. Let's talk about the cytoskeleton, and then we'll wrap up this chapter. The cytoskeleton is responsible for directed cell movements, and then we'll go on to our second Harvard textbook. Maybe we'll do linear algebra next, or maybe computer science. I don't know, whatever you guys want. The cytoskeleton is responsible for directed cell movements. The cytosol is not just a structureless soup of chemicals and organelles. Using an EM, one can see that in eukaryotic cells, that the cytosol is crisscrossed by long, fine filaments. Frequently, the filaments are seen to be anchored at one end of the plasma membrane or to radiate out from a central site adjacent to the nucleus. This system of protein filaments is called the cytoskeleton. And it's composed of 3 major filament types. This is pretty important. The thinnest of these filament types are actin filaments. They're abundant in all eukaryotic cells, but occur in especially large numbers inside muscle cells, where they serve as an essential part of the machinery responsible for contraction. The thickest filaments in the cytosol are called microtubules, really important, because they have the form of minute hollow tubes. In dividing cells, they become reorganized into a spectacular array that helps pull the duplicated chromosomes apart. and distribute them equally into the two daughter cells. Intermediate in thickness between actin and microtubules are the intermediate filaments. And I want you to think about that word filament, because it will come up again and again, which serve to strengthen most animal cells. So without these proteins, we would have cells that are just too floppy to really do much. These three types of filaments, together with other proteins that attach to them, form a system of girders, ropes, and motors that give the cell its mechanical strength, control its shape, and drives and guides its movements. That's really, really important. Without these, the description of the cell is completely incomplete. All right, the cytoskeleton is a network of protein filaments that can be seen crisscrossing the cytoplasm of eukaryotic cells. Three major types of filaments can be detected through fluorescent stains. So here's the actin, here's the microtubules, and here are intermediate filaments. Filaments are not found in the cytoplasm of cells with cell walls, such as plant cells. They have their own structure. Let's look at microtubules, which help segregate the chromosomes in a dividing animal cell. This is a TEM and schematic drawing here and here that show duplicated chromosomes attached to the microtubules of a mitotic spindle, which we'll talk about later on. When a cell divides, its nuclear envelope breaks down, and its DNA condenses into visible chromosomes, each of which has duplicated to form a pair of conjoined chromosomes that will be pulled apart into separate daughter cells by the spindle microtubules. But microtubules go well beyond just cell division. Because the cytoskeleton governs the internal organization of the cell as well as its external features, it is necessary to a plant cell, boxed in by a tough cell wall, as it is to an animal cell that freely bends, stretches, swims, or crawls. In a plant cell, organelles such as mitochondria are driven by the constant stream around the cell interior along cytoskeletal tracts, and in animal and plant cells alike depend on the cytoskeleton to separate their internal components into two daughter cells during cell division. The cytoskeleton's role in cell division may be its most ancient function. Even bacteria contain proteins that are distantly related to those that form the cytoskeletal elements involved in our cell division. In bacteria, these proteins also form filaments that play a part in cell division. We examine the cytoskeleton in Chapter 17, discuss its role in cell division in 18, and review how it responds to signals from the outside of the cell in 16. So anyway, the cytosol is far from static. The cell interior is in constant motion. The cytoskeleton is a dynamic jungle of protein ropes that are continually being strung together and taken apart. Its filaments can assemble and then disappear in a matter of minutes. Motor proteins use the energy stored in molecules of ATP to trundle along these tracks and cables, carrying organelles and proteins throughout the cytoplasm and racing across the width of the cell in seconds. In addition, large and small molecules that fill every free space of the cell are knocked to and fro by random thermal motion, constantly colliding with one another and with other structures in the cell's crowded cytosol. Of course, neither the bustling nature of the cell's interior nor the details of the cell structure were appreciated when scientists first peered at cells in a microscope. Our knowledge of cell structure accumulated slowly. Let's look at the historical landmarks. 1665, we know that Hooke used a primitive microscope to look at cork, and he named them cells. 1674, Leeuwenhoek reports his discovery of protozoa. He sees bacteria for the first time. Later, Brown publishes his microscopic observation of orchids, describing the cell nucleus. Schleiden and Schwann proposed cell theory in 1839. 1857, Kolliker describes mitochondria. 1879, Fleming describes chromosome behavior and mitosis. 1881, Kyle, who's extremely important name in biology, and other histologists developed staining methods. Golgi sees the first season describes the Golgi apparatus in 1898, really crucial. Boveri links chromosomes to heredity by observing chromosome behavior during sexual reproduction. Palade, Porter, and Sjöstrand developed methods of EM that enable many intracellular structures to be seen for the first time. In one of the applications, Huxley shows that muscle contains arrays of protein filaments. 1957, Robertson describes the bilayer structure of the cell membrane, really important, seen for the first time in an EM. So that was not that long ago, if you think about it, about 70 years ago. 1960, Kendrew describes the first detailed protein structure, sperm whale myoglobin, to a resolution of just 0.2 nanometers, very impressive, using X-ray crystallography. So that was 1960. And then Perutz proposes an even lower resolution structure for hemoglobin. 1965, a cell fractionation technique separates different organelles. 1968, Petran makes the first confocal microscope. 1970, Fry and Eddy used fluorescence antibodies, and that shows that molecules can get across the cell membrane. 1974, fluorescent antibodies used to stain the cytoskeleton. 1994, fairly recent, after I was born at least, they introduced GFP. GFP is a very important, very, very important innovation. 1990s, these folks developed techniques for super resolution fluorescence microscopy. So this is a relatively new technique. So let's go on to eukaryotic cells. Many may have originated as predators. Eukaryotic cells are typically 10 times the length and 1,000 times the volume of prokaryotic cells, although there's a huge size variation. They also possess a huge collection of features, a whole collection of features: nucleus, cytoskeleton, mitochondria, organelles that set them apart from bacteria and archaea. When and how eukaryotes evolved these systems remains something of a mystery. Although the eukaryotes, bacteria, and archaea must have diverged from one another very early in the history of life, eukaryotes did not acquire all their distinctive features at the same time. That makes sense. According to one theory, the ancestral eukaryotic cell was a predator that fed by capturing other cells. Such a way of life requires a large size, flexible membrane, and a cytoskeleton to help the cell move and eat. The nuclear compartment may have evolved to keep the DNA segregated. We should discuss the relative advantages and disadvantages of light and electron microscopy. Obviously, the disadvantage of electron is the preparation. The disadvantage of light is the resolution size. How could you best visualize a living skin cell, yeast, mitochondria, bacteria, and microtubule? All right, so the nuclear compartment evolved to keep the DNA separate from the hurly-burly, so as to allow more delicate and complex control of the way the cell reads out its genetic information. Such a primitive eukaryotic cell with nucleus and cytoskeleton was most likely the sort of cell that engulfed the free-living, oxygen-consuming bacteria that were likely the ancestors of the mitochondria. This partnership is thought to have been established 1.5 billion years ago, when the Earth's atmosphere first became rich in oxygen. A subset of these cells later acquired chloroplasts for engulfing photosynthetic bacteria. The likely history of these endosymbiotic events is illustrated above. That single-celled eukaryotes can prey upon and swallow other cells is born out of the behavior of many present-day protozoans, a class of free-living modal unicellular organisms. Protozoans, very important. Didinium, for example, is a large carnivorous, I don't know if carnivorous is the right word, protozoan with a diameter of about 150 micrometers. Fairly large, 10 times that of the average human cell. It has a globular body encircled by 2 fringes of cilia, and its front end is flattened except for a single protrusion rather like a snout. Didinium swims at high speed by means of its beating cilia. This thing ugly? And it's eating this thing. When it encounters a suitable prey, usually another type of protozoan, it releases numerous small paralyzing darts from its snout region. Didinium then attaches to and devours the other cell, inverting like a hollow ball to engulf its victim, which can be almost as large as itself. Amazing. See how much more we have to go in this chapter. This is longer than I expected. Do we want to try a different textbook? This is just chapter 1. We're almost done. Let's see, it's 12:30. When did we start? Let's take a break from biology. We have a few other textbooks to go through. Let me see what I got cooking. I've got statistics, unlocking the power of data. These are all Harvard textbooks that are currently used in the Harvard curriculum. I don't want a signature panel. So this is a subject a lot of people don't like. There's a more fun book called Hacker's Delight. I'm not sure what it's about, but it's a suggested CS book. I probably should read that. It looks very intense. There's another book called Programming in C. There's another book called How Computers Work. These are the suggested books for CS50, Harvard's class. There's General Chemistry. We have Linear Algebra with applications. So we've got a choice of about 5 or 6 different books. I think Hacker's Delight looks pretty cool. So this reminds me of a book called Code by Petzold. Second edition by Henry Warren Jr., published by Addison Wellesley. It's a little bit of an older book. This looks very advanced. All right, this is a forward. When I first got a summer job at MIT's Project MAC almost 30 years ago, I was delighted to be able to work with the DEC PDP-10 computer, which was more fun to program in assembly language than any other computer, bar none, because of its rich yet tractable set of instructions for performing bit tests, bit masking, field manipulation, and operations on integers. Though the PDP-10 has not been manufactured for quite some years, there remains a thriving cult of enthusiasts who keep old PDP-10 hardware running and who run old PDP-10 software, entire operating systems in their applications, by using personal computers to simulate the PDP-10 instruction set. They even write new software. There is now at least one website with pages that are served up by simulated PDP-10. Come on, stop laughing. It's no sillier than keeping antique cars running. I also enjoyed in that summer of 1972 reading a brand new MIT research memo called HackMem, a bizarre and eclectic potpourri of technical trivia. The subject matter ranged from electric circuits to number theory, but what intrigued me most was its small catalog of ingenious little programming tricks. Each such gem would typically describe some plausible yet unusual operation on integers or bit strings, such as counting the 1 bits in a word. A word is a technical term. That could easily be programmed using either a longish fixed sequence of machine instructions or a loop, and then show how the same thing might be done much more cleverly, using just 4 or 3 or 2 carefully chosen instructions whose interactions are not at all obvious until explained or fathomed. For me, devouring these little programming nuggets was like eating peanuts, or rather bonbons. I just couldn't stop. And there was a certain richness to them, a certain intellectual depth, elegance, even poetry. Surely, I thought, there must be more of these, and indeed, over the years I collected and in some case discovered a few more. There ought to be a book of them. I was generally thrilled when I saw Hank Warren's manuscript. He has systematically collected these little programming tricks, organized them thematically, and explained them clearly. While some of them may be described in terms of machine instructions, this is not a book only for assembly language programmers. The subject matter is basic structural relationships among integers and bit strings in a computer and efficient techniques for performing useful operations on them. These techniques are just as useful in the C or Java programming languages as they are in assembly. Why HackMem? Well, it's short for Hack's Memo. One 36-bit PDP-10 word could hold 6 6-bit characters. So a lot of the names PDP-10 hackers worked with were limited to 6 characters. We were used to glancing at a 6-character abbreviated name and instantly decoding the contractions. So naming the memo hackmem made sense at the time, at least to the hackers. Many books and algorithms in data structure teach complicated techniques for sorting and searching, for maintaining hash tables and binary trees, for dealing with records and pointers. They overlook what can be done with very tiny pieces of data, bits and arrays of bits. It is amazing what can be done with just binary addition and subtraction and maybe some bitwise operations. The fact that the carry chain allows a single bit to affect all the bits to its left makes addition a peculiarly powerful data manipulation operation in ways that are not widely appreciated. It's funny, when I learned the bitwise operators in JavaScript, I was confused as to why anybody would ever need such a thing. But, you know, it's a different time and a different era. Yes, there ought to be a book about these techniques. Now it's in your hands, and it's terrific. If you write optimizing compilers or high-performance code, you must read this book. You otherwise might not use this bag of tricks every single day, but if you find yourself stuck in some situation where you apparently need to loop over the bits in a word or to perform some operation on integers, it just seems harder to code than it ought, or you really need the inner loop of some integer or bit fiddly computation to run twice as fast, this is the place to look. Or maybe you'll just find yourself reading it straight through out of sheer pleasure. Guy Steele, Burlington, Mass., 2002. Right, this is the preface. Caveat emptor, the cost of software maintenance increases with the square of the programmer's creativity. First law of programming creativity, Robert Bliss, 1992. This is a collection of small programming tricks that I've come across over many years. Many of them will work only in computers that represent integers in 2's complement form. Important concept here. Although a 32-bit machine is assumed when the register length is relevant, most of the tricks are easily adapted to machines with other register sizes. Oh boy, this is complicated. This book does not deal with large tricks such as sophisticated sorting and compiler optimization techniques. Rather, it deals with small tricks that usually involve individual computer words or instructions, such as counting the number of 1 bits in a word. Such tricks often use a mixture of arithmetic and logical instructions. It is assumed throughout that integer overflow interrupts have been masked off so they cannot occur. C, Fortran, and even Java programs run in this environment, but Pascal and Ada users beware. The presentation is informal. Proofs are given only when the algorithm is not obvious, and sometimes not even then. The methods use computer arithmetic, floor functions, mixtures of arithmetic and logical operations, and so on. Proofs in this domain are often difficult and awkward to express. To reduce typos, many of the algorithms have been executed. This is why they're given in a real programming language, even though, like every computer language, it has some ugly features. C is used for the high-level language because it's widely known. It also the straightforward mix of integer and bit string operations, and C compilers that produce high-quality object code are available. Occasionally, machine language is used, employing a 3-address format, mainly for the ease of readability. The assembly language used is that of a fictitious machine that is representative of today's RISC computers, reduced instruction set code. Branch-free code is favored because in most computers, branches slow down instruction fetching and inhibit executing instructions in parallel. Another problem with branching is that they can inhibit compiler optimization such as instruction scheduling, commenting, and register allocation. That is, the compiler may be more effective at these optimizations with a program that consists of a few large basic blocks rather than many small ones. The code sequences also tend to favor small, immediate values, comparisons to 0 rather than to some other number, and instruction-level parallelism. Although much of the code would become more concise by using table lookups for memory, this is not often mentioned. This is because loads are becoming more expensive relative to arithmetic instructions, and the table lookup methods are often not very interesting, although they are often practical. But there are exceptional cases. Finally, I should mention that the term hacker in the title is meant in the original sense of an aficionado of computers, someone who enjoys making computers do new things or do old things in a new and clever way. The hacker is usually quite good at his craft, his or her craft, but may very well not be a professional computer programmer or designer. The hacker's work may be useful or may be just a game. As an example of the latter, more than one determined hacker has written a program which, when executed, writes out an exact copy of itself. This is the sense in which we use the term hacker. If you're looking for tips on how to break into somebody else's computer, you won't find them here. One such program written in C is main car star p equals main car star p, some escape characters, void printf, void printf. So I don't know old C that well. I forgot what the asterisk operator in old C does. Let's take a look. Well, obviously it's a multiplier, but for dereference operator. Okay. I think one decent goal is to understand this program. We will try to understand this program maybe now, maybe at a later date, but I think this would be pretty interesting. I mean, it just seems to print out pretty simple. It just prints out P, and P is this thing. So that's the basic sense of it, right? It's a string, right? So it's like it used to have to, like car 10 or something like that, used to have to enumerate how many like a string is actually just an array of cars in old C, if I recall correctly. All right, anyway. All right, first I want to thank Bruce Shriver and Dennis Allison for encouraging me to publish this book, indebted to many colleagues at IBM, several of whom are cited in the bibliography. Martin Hopkins, who I think of as Mr. Compiler at IBM, has been relentless in his drive to make every cycle count, and I'm sure some of his spirit has rubbed off on me. Guy Steele completed a 50-page review that includes new subject areas to address, such as bit shuffling and unshuffling, the sheep and the goats operation, and many others. He suggested algorithms that beat the ones I used. He was extremely thorough. For example, I had erroneously written that the hexadecimal number AAA factors as 2 times 3 times 17 times 257 times 65537. Guy pointed out that the 3 should be a 5. He suggested improvements, etc., etc. Wherever you see parallel prefix in this book, the material is due to Guy. All right, so this is Yorktown, New York, where IBM is still located. All right, let's have some fun. Chapter 1, Introduction. 1.1 Notation. This book distinguishes between mathematical expressions of ordinary arithmetic and those that describe the operation of a computer. In computer arithmetic, operands, it's not a word that's not used that much, but it's still fundamental. Operands are bit strings or bit vectors of some defined fixed length. Expressions in computer arithmetic are similar to those of ordinary arithmetic, but the variables denote the contents of the computer registers. The value of a computer arithmetic expression is simply a string of bits with no particular interpretation. An operator, however, interprets its operands, right? These are arguments for the operator. So if you have, quite simply, really easy stuff, 2 times 3, the multiplication is the operator, and these are the operands. Right? And I just think sometimes we want to be able to refer to the language abstractly or generally. And there are specific subtypes of operands such as, right, a variable. What's the operand like this? What do we call that? Sometimes called a static? Or maybe another better word for it? These are subtypes of operands. We can pass a lot of operands to an operator. So just getting this semantics down. All right. For example, a comparison operator, what's a comparison operator? Greater than, less than, equal, or equal, might interpret its operand as signed binary integers or as unsigned binary integers, positive or negative, right? Our computer arithmetic notation uses distinct symbols to make the type of comparison clear. Let's see here. The main difference between computer arithmetic and ordinary arithmetic is that in computer arithmetic, the results of addition, subtraction, multiplication are reduced modulo 2 to the n. What does that mean? where n is the size of the word in the machine. So I think we're referring to binary math here. Another difference is that computer arithmetic includes a large number of operations. In addition to the 4 basic arithmetic operations, computer arithmetic includes the logical and, exclusive or, XOR, compare, shift left, and so on. Unless specified otherwise, the word size is 32 bits, and side integers are represented in 2's complement form. So these are all, depending on how you look at them, advanced or basic stuff in computing. Expressions of computer arithmetic are written similarly to those of ordinary arithmetic, except that the variables that denote the contents of the registers are in boldface type. This convention is commonly used in vector algebra. We regard a computer word as a vector of single bits. Constants also appear in boldface type when they denote the contents of a computer register. This has no analogy with vector algebra because in vector algebra the only way to write a constant is to display the vector's components. When a constant denotes part of an instruction, such as the immediate field of a shift instruction, lightface type is used. If an operator such as plus or addition has boldface operands, then the operator denotes that the computer's addition operation, vector addition. If the operands are lightface, then the operator denotes the ordinary scalar. By the way, that's the word I was looking for here. These are scalars. When you have just a static number like that, sometimes it's called a scalar. We use a lightface variable x to denote the arithmetic value of the boldfaced variable x under an interpretation, signed or unsigned, that should be clear from the context. Thus, if x equals 0x80000000 and y equals the same, then under signed integer interpretation, x equals y equals negative 2 to the 31st. X plus y equals negative 2 to the 32nd, and x plus y equals 0. Here, x, 0x, 8000000 is hexadecimal notation for a bit string consisting of a 1 bit followed by 31 0 bits. Hope you get that. I don't. Bits are numbered from the right, with the rightmost least significant bit being bit 0. The term bits, nibbles, bytes, half-words, words, and double words refer to lengths 1, 4, 8, 16, 32, and 64 bits. So this was my favorite word growing up studying computers. Nibbles. Nibble is half a byte. Because, a byte is a big byte, so a nibble is half a byte, right? What's smaller than a byte? A nibble. Short and simple sections of code are written in computer algebra using its assignment operator, the left arrow, and occasionally an if statement. In this role, computer algebra is serving as little more than a machine-independent way of writing assembly language code. Programs too long or complex for computer algebra are written in the C programming language as defined by the ISO 1999 standard. A complete description of C would be out of place in this book, but Table 1.1 contains a brief summary of most of the elements of C that are used herein. This is provided for the benefit of the reader who's familiar with some of the procedural programming language, but not with C. Table 1.1 also shows the operators of our computer algebraic arithmetic language. Operators are listed from highest precedence, or tightest binding, to lowest. In the precedence column, L means left associative. So if you have two operators, they'll associate the first two operands, and right means right associative, which will do the opposite. Our computer algebraic notation follows C in precedence and associativity. In addition to the notations described in 1.1, those of Boolean algebra and standard mathematics are used in explanations where necessary. So let's look at this, the expressions of C in computer algebra. If this doesn't blow your mind, I don't know what will. So here's logical not. I recognize this immediately. This is arithmetic negation, very simple. So not just reverses a 0 to a 1 or a 1 to a 0. This is a bitwise not, which is different. This is the ones complement, so this is just 1 bit. We have an exponent, type conversion, so this is coercion of type, pre-increment and pre-increment, and decrement, post-increment and decrement. So this is familiar in most languages. You have the negative of the absolute value. So take the absolute value and make a negative. You have the absolute value. But the absolute value of negative 2 to the 31st is still negative 2 to the 31st. Okay? We have functions, which are the highest binders. And different variables are bit selection. And the kth component of an array. And then hexadecimal and binary constants, which are the highest precedence. And we get to lower and lower precedence. We have multiplication and division. We have signed integer division, unsigned integer division. We have multiplication, which is modulo the word size. We have modulo or remainder. Let's see. Again, remainder, x reduced modulo of y. Addition, subtraction. Now we have the confusing ones. Shifts. So shift left, shift right, which we'll need to explain some other time. Signed comparisons, pretty simple. Is something bigger than or less than? Is it equal to? Double equals is equal to. Equals is the assignment operator, which we'll get to in a second. Bitwise exclusive, bitwise equivalence, bitwise or. So this is different from these comparison operators. Conditional and, conditional or, concatenate, and here's assignment, which is the lowest precedence. Keep that in mind. Assignment has the lowest precedence. Our computer algebra uses other functions in addition to absolute, remainder, and so on. These are defined when introduced. In C, the expression x less than y less than z means to evaluate x less than y to a 0-1 valued result, a binary, a Boolean, I should say, and then compare that result to z. In computer algebra, this expression means x is less than y and y is less than z. C has 3 control loop statements, while, do, and for. The while statement is while this statement. The first expression is evaluated. If it's true, statement is executed, and control returns to evaluate it again. If it's false, the while loop terminates. So do is similar, except the test is at the end. And for has the typical for structure. So I don't know why I'm skipping this so quickly, but this is a lot of fun because I wanted to get to here, which is the instruction set. And again, this is very technical stuff. This is not the required textbook for CS50. This is a suggested reading. Again, this is basically machine code and how to go through machine code. So I guess I was going through that table quickly because I didn't want to belabor every little piece of the table, even though it is probably pretty important. So I'll just finish this chapter and we can close this book and we can say at least that we read chapter 1, right? All right, C has 3 loop control statements, while, do, and for. The while statement is written, while expression, do this statement. So first you evaluate the expression. If it's true, the statement is executed and the control returns to evaluate the expression again. So that's why it's a loop. And if the expression is false, the loop is over. The do statement is similar, except the test is at the bottom. So you do this statement while this is true. So the statement is executed first. So the difference between do and while is you guarantee at least one statement execution in do. Then you evaluate the expression. If it's true, the process is repeated, and if it's false, the loop will terminate. The for statement, which we've all hopefully written one for statement in our life, takes these 3 variables. So you have E1, which is the assignment statement. So usually you assign like some counter for I equals 1 or 0 or whatever. E2 is the comparison. So this is the kind of like test. So as long as I is less than n, this bunch of code will continue. And then at the end of it all, add 1 to I. So this way at least I will start to approach n. It may approach it quickly, it may approach it slowly, but it will approach it at some point. And there are plenty of examples. I have, just a simple formula in here, and you can see already a couple of for loops. So for loops have never really have been around for the test of time, since the test of time. This is one of the few contexts in which we use the post increment operator. The ISO-C standard does not specify whether right shifts, this is the right shift operator, of sign quantities are 0 propagating or sign propagating. This is very advanced, low-level computer programming, which is not really done these days anymore, at least for most people. In the C code herein, it is assumed that if the left operand is signed, then a sign propagating shift results, and if it is unsigned, then a zero propagating shift results, following ISO. Most standard C compilers work this way. It is assumed that left shifts are logical. Some machines, mostly older ones, provide an arithmetic left shift, in which the sign bit is retained. Another potential problem with shifts is that the ISO C standard specifies that if the shift amount is negative or is greater than or equal to the width of the left operand, the result is undefined. But nearly all 32-bit machines treat shift amounts modulo 32 or 64. The code herein relies on one of these behaviors. An explanation is given when the distinction is important. All right, this is the cool part. Section 1.2, chapter 1.2, instruction set and execution time model. To permit a rough comparison of algorithms, we imagine them being coded for a machine with an instruction set similar to that of today's general-purpose RISC computers, such as the IBM RS-6000, the Oracle SPARC, and the ARM architecture. The machine is 3-address and has a fairly large number of general-purpose registers, that is, 16 or more. Unless otherwise specified, the registers are 32 bits long. General register 0 contains a permanent 0, and the others can be used uniformly for any purpose. In the interest of simplicity, there are no special purpose registers, such as a condition register or a register to hold status bits, such as overflow. The machine has no floating point instructions. Floating point is only a minor topic in this book, being confined to chapter 17. We recognize two varieties of RISC, a basic RISC having the instructions shown in 1.2, and a full RISC, having all the instructions of the basic RISC plus those in 1.3. So what's basic RISC? I used to be infatuated with this stuff as a kid because the video game systems I played on had RISC chips, and I always wanted to know what exactly was a RISC chip. So here are the opcodes: add, sub, mul, div, divu, rem, and remu. These sound pretty obvious what they are. The operands they take, RT, RA, and RB. So RT, RA, op, RB, where op is add, subtract, multiply, divide sign, divides unsigned, remainder unsigned, or remainder signed or remainder unsigned. So RA, op, RB will give you RT. ADDI and MULI. Op is add or multiply, and I is a 16-bit signed immediate value. Add is, this is a shift, and or XOR, I'm confused by what this is. I think you're adding a immediate signed value, but I'm not sure. So this is bitwise and or XOR. This is a same thing, except one of the operands is an unsigned immediate value. Here are some more BEQ, BNE, BLT, BLE, BGT, or BGE. These are branches. So BT and BF are branches as well. Branch true, branch false. So what? Branch equals, less than, greater than or equal to, less than or equal to, branch not equal to. So these are like if statements. How would you like to program like this? Now we have CMP, EQ, CMP, NE, LT, LE, GT, GE, LTU, LEU, GTU, and GEU. These are, again, comparisons. Compare for equality, inequality, less than, and so forth. So these are really old assembly tools. See, compare again, except there's a 16-bit signed immediate value. Compare again, except there's a second comparand. I love that word. Not an operand, but a comparand. Phew. All right, this is loading into a memory location or register. Here's a multiplication again. Here's not. Here's the shift. The shift, I guess, what are we calling these again? Opcodes. And then store into memory. All right, so in these tables, RA and RB appearing as source operands really means the contents of those registers. A real machine would have branch and link for subroutine calls, branch to the address containing register for subroutine returns and switches, and possibly some instructions for dealing with special purpose registers. It would, of course, have a number of privileged instructions and instructions for calling on supervisor services. It might also have floating point instructions. Some other computational instructions that a RISC computer might have are identified in 1.3. Those are discussed in later chapters. It is convenient to provide the machine's assembler with a few extended mnemonics. These are like macros, whose expansion is usually a single instruction. Some possibilities are in 1.4. The load immediate instruction expands into 1 or 2 instructions, as required by the immediate value I. For example, if 0 is less than or equal to I, which is less than or equal to 2 to the 16th, and or immediate, ORI, from R1 can also be used. If negative 2 to the 15th is less than or equal to I and less than 0, an add immediate from R0 can be used. If the rightmost 16 bits of I are 0, add immediate shifted, ADDIS, that's add immediate shifted, now I understand, can be used. Otherwise, 2 instructions are required, such as add immediate shifted followed by or I. Alternatively, in the last case, a load for memory can be used, but for execution time and space estimates, we assume that 2 elementary arithmetic instructions are used. Of course, which instructions belong in the basic RISC and which belong in full RISC is very much a matter of judgment. Quite possibly, divide, unsigned, and remainder instructions should be moved to full RISC. Conversely, possibly load byte signed should be in the basic RISC category. It is in the full RISC set because it's probably of rather low frequency of use, and because in some technologies it is difficult to propagate a sign bit through so many positions and still make cycle time. The distinction between basic and full RISC involves many other questionable judgments, but we don't need to dwell on them. The instructions are limited to 2 storage registers and 1 target, which simplifies the computer. The register file requires no more than 2 read ports and 1 write port. It also simplifies an optimizing compiler, because the compiler does not need to deal with instructions that have multiple targets. The price paid for this is that the program that wants both the quotient and remainder of 2 numbers, which is not uncommon, must execute 2 instructions, divide and remainder, and this still plagues JavaScript and C to this day. The usual machine division algorithm produces the remainder as a byproduct, so many machines make them both available as a result of 1 execution of divide. Similar remarks apply to obtaining the double word products of 2 words. The conditional move instructions, move Q, ostensibly have only 2 source operands, but in a sense they have 3. Because the result of the instruction depends on the values of RT, RA, and RB, the machine executes instructions out of order. A machine that executes instructions out of order must treat RT in these instructions as both a use and a set. That is, an instruction that sets RT followed by a conditional move that sets RT must be executed in that order, and the result of the first instruction cannot be discarded. Thus, the designer of such a machine may elect to omit the conditional move instructions to avoid having to consider an instruction with logically 3 source operands. On the other hand, conditional move instructions do save branches. Instruction formats are not relevant for the purposes of this book, but the full RISC instruction set described above, with floating point, and a few supervisory instructions added, can be implemented with 32-bit instructions on a machine with 32 general purpose registers, 5-bit register fields. By reducing the immediate fields of compare, load, store, and trap instructions to 14 bits, the same holds for a machine with 64 general purpose registers. Wow, 6-bit register fields. All right, execution time. We assume that all instructions execute in 1 cycle, except for the multiply, divide, and remainder instructions, for which we do not assume that any particular execution time. Branches take 1 cycle whether they branch or fall through. The load immediate instruction is counted as 1 or 2 cycles depending on whether 1 or 2 elementary arithmetic instructions are required to generate the constant in a register. Although load and store instructions are not often used in this book, we assume they take 1 cycle and ignore any load delay, a time lapse between when a load instruction completes in the arithmetic unit and when the requested data is available for a subsequent instruction. However, knowing the number of cycles used by all the arithmetic and logical instructions is often insufficient for estimating the execution time of a program. Execution can be slowed substantially by load delays and by delays in fetching instructions. These delays, although very important, are increasing in importance and are not discussed in this book. Another factor, one that improves execution time, is what is called instruction-level parallelism, which is found in many contemporary RISC chips, particularly those for high-end machines. These machines have multiple execution units and sufficient instruction dispatching capability to execute instructions in parallel when they are independent, that is, when neither uses the result of another, and they don't both set the same register or status bit. Because this capability is now quite common, the presence of independent operations is often pointed out in this book. Thus, we might say that such and such formula can be coded in such a way that it requires 8 instructions and executes in 5 cycles on a machine with unlimited instruction-level parallelism. This means that if the instructions are arranged in a proper order or scheduled, a machine with sufficient number of adders, shifters, logical units, and registers can, in principle, execute the code in 5 cycles. We don't make too much of this because machines differ greatly in their instruction-level parallelism capabilities. For example, an IBM RS-6000 processor from around 1992 has a 3-input adder and can execute 2 consecutive add type instructions in parallel, even when one feeds another. In other words, in add feeding a compare or the base register of a load. As a contrary example, consider a simple computer, possibly for low-cost embedded application, that has only one read port on its register file. Normally, this machine would take an extra cycle to do a second read of the register file for an instruction that has 2 register input operands. However, suppose it has a bypass so that if an instruction feeds an operand of the immediately following instruction, then that operand is available without reading the register file. On such a machine, it is actually advantageous if each instruction feeds the next. That is, if the code has no parallelism. Okay, can you express the for loop in terms of a while loop? Can it be expressed as a do loop? That's another very interesting question. This is basic computer science. Code a loop in C in which the unsigned integer control variable I takes on all the values from 0 to and including the maximum unsigned number 0xFFF FFFF on a 32-bit machine. For the more experienced reader, the instructions of the basic and full risks defined in this book can be executed with at most 2 register reads and 1 write. What are some of the common or plausible RISC instructions that either need more source operands or need to do more than 1 register write? Cool. So that is the introduction. There's some extended mnemonics here I want to go through really quickly. 1.3 is full RISC, so we have absolute and negative absolute. We have bitwise and with a complement, equivalence, negation, and, or negative, or NAND, not and, or negate and or with complement. We have bit extraction with sign fill. We have insertion. We have number of leading zeros, NLZ. We have pop, just how many 1 bits are in something. We have loading assigned byte. We have move if there's equal or other conditionals. We have shifts, more shifts with certain amounts. And we have trap if there's a certain situation or conditional, and other traps. And then we have this little extended mnemonic, which is an unconditional branch, load immediate, move, this is a very common, very, very common assembly instruction. Move register RA to RT, so this takes 2 operands. Negate with 2's complement, and subtract immediate. So that's chapter 1 of this book. It's called Hacker's Delight. It's a suggested reading for CS50, which is the Harvard undergraduate course. There are 3 suggested readings. This is just one of them. I hope you enjoyed that. I'm going to take a 2-minute break to eat and maybe just pause. I'm going to play chess in the meantime, so I hope you enjoy that. And then we'll get to textbook 3. English opening here. I don't mind that at all. Good morning, good morning. I don't know what to do. I might play one of the Botez sisters. I have to practice though. Fuck. Okay. Easy win.
Goddamn. It's those extra seconds I took to clean up. Personally, I think it's this kitty's fault right here. You see her? You see her? What are you lounging around? Why don't you do some work? That's her favorite little chair to sleep in. Why don't you do some work, huh? I'll put you to work. How would you like to be involved in the CRISPR experiment? Prepare the conditioning regimen. Stronger, faster, we can rebuild her. I got her yesterday. One more chess game. We go back to the Harvard curriculum. I really want to learn bitwise operators. I doubt anyone at Harvard CS50 actually learns that, by the way. Hacker's delight. Hacker's delight. Oh, delight that. Jeremy, let's go play water polo. Delight this. No. Look at these nerds. Let's go rowing. She is like a little coffee. MSI? Well, we're building a lot of good software for MSI, so I'm not too worried about that. These things take time. Sometimes takes many years. But the cat isn't too valuable. My dad thinks cats are really valuable. Cats are really smart. Nope. Completely untrue. Cats are useless. Oh, here's the sack. Look at this shit. Oh, you think you're tough. Oh, you think you're bad. Well, you are bad, I'll say that. There's a couple ways to play this, right? Okay. Right here? What do you think? Right here. Knight's coming there. That just leads to mate. Well. What's wrong? You sneeze? You sneeze, you sneeze. Are you a little beast? You got a cold? You're sneezing? I hate you, kitty. I hate you. I'm gonna eat you later. You're gonna be delicious. Yeah. Kitty stew. Nom 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 nom. I'm gonna eat some kitty stew. I'm gonna put you in the pot. I don't care what they say on YouTube. I don't care. You're going in the pot. I'm hungry. Yeah.
I'm fattening you up with those treats. Don't look at me like that. I may have miscalculated. Fuck. Alright, how many grading points I want to lose today? Oh. You forgot about Dre. We don't have a name yet. If you guys want to... My dad said that we should leverage the strength of the community to name her. So if you guys have a good name, it's wide open. Jelly bean, cappuccino, or cupcake. You're like my sister with these stupid names. She doesn't deserve a name like that. Ugh, that's a good move. She deserves a name like Ugly. Shit. Fuck. No, nothing cute to make up for trashy. You're KDR, you're talking just like my sister. No. She deserves a terrible name. Like Ugly or Mid or she's actually got a little bit of a running impediment. She's like clumsy. I like that word. I like the name, clumsy. Mid. Kamala. Name her Ugly Trash Can. Worst cat of all time. That's her name. Piece of trash. Takes. Check. I'm trying to figure out if I can pull this sacrifice off. Check. Probably get a... I'm trying to see if I can get a draw by perpetual. Schrodinger. I'm not naming her anything. She doesn't even deserve a name. Okay, here we go. Takes. Check. Yeah, that's perpetual. Take the perpetual or play for the win. Imagine it's not perpetual. Undefined, I love that. Null. I set her to null. All right, so enough about bitwise operators. This is another book for Harvard. This is a lot simpler, it looks like. It's not that simple. This also reminds me of Code by Petzold. Well, this won't take us long to read, so I recommend we read this. This is called How Computers Work. It's part of the CS50 recommended books by Ron White. National bestseller, more than 2 million copies sold. Okay, or of this series at least. There's a transistor, or a resistor. I think that's a resistor actually. Anyway, this is the 10th edition of this thing. There's little gremlins inside the computer. All right, The Evolution of Technology, 10th edition. Remember, Harvard book, 2015. For Shannon and Michael, who always asked why, and Sue, who asked why not. Ron White has been writing How Computers Work for 20 years. He's also been an executive editor at PC Computing, Byte, and GroovyPost.com. Computer columnist for Windows Source and 80 Micro. How Computers Work was named the best nonfiction computer book, and his writing's been honored by this and that. I'm going to skip the acknowledgements. Famous quote, any sufficiently advanced technology is indistinguishable from magic, often attributed to Isaac Asimov, but it's Arthur C. Clarke, who is his predecessor, I believe. Sorcerers have their magic wands, powerful, potentially dangerous tools with lives of their own. Witches have their familiars, creatures disguised as household beasts that could, if they choose, wreak the richest havoc. Mystics have their golems, beings built of wood and tin, brought to life to do their master's bidding. We have our personal computers. PCs, too, are powerful creations that often seem to have a life of their own. Usually they respond to a wave of a mouse or a spoken incantation by performing tasks we couldn't imagine doing ourselves without some sort of preternatural help. But even as computers successfully carry out our commands, it is often difficult to quell the feeling that there's some wizardry at work here. And then there are times when our PCs, like malevolent spirits, rebel and open the gates of chaos onto our neatly ordered columns of numbers, our carefully wrought sentences, and our beautifully crafted graphics. When that happens, we're often convinced that we are indeed playing with power not entirely under our control. We become sorcerers' apprentices, whose every attempt to write things leads to deeper trouble. Whether our computers are faithful servants or imps, most of us soon realize there's much more going on inside these solid boxes than we really understand. PCs are secretive. Open their tightly sealed cases and you're confronted with poker-faced components. Few give any clues as to what they're about. Most of them consist of sphinx-like microchips that offer no more information about themselves than some obscure code printed on their impenetrable surfaces. The maze of circuit tracings etched on the boards is fascinating but meaningless hieroglyphics. Some crucial parts, such as the hard drive or power supply, are sealed with printed omens about the dangers of peeking inside, omens that put to shame the warnings on a pharaoh's tomb. This book is based on two ideas. One is that the magic we understand is safer and more powerful than the magic we don't. This is not a hands-on how-to book. Don't look for any instructions for taking a screwdriver to this part or the other. But perhaps your knowing more about what's going on inside of all these stoic components make them a little less formidable than when something does go awry. The second idea behind this book is that knowledge in itself is a worthwhile and enjoyable goal. How about that? This book is written to respond to your random musings about the goings-on inside the box you sit in front of several hours a day. If this book puts your questions to rest or raises new ones, it will have done its job. At the same time, however, I'm trusting that knowing the secrets behind the magician's legerdemain won't spoil the show. This is a real danger. Mystery is often as compelling as knowledge, and I hate to think that anything you read in this book takes away that sense of wonder you have when you first manage to make your PC do some grand new trick. I hope that instead this book makes you a more confident sorcerer. All right, if automobiles had followed the same development cycles as the computer, a Rolls-Royce would today cost $100, get 1,000,000 miles per gallon, and explode once a year, killing everyone inside. Robert Kringley. Introduction to the 10th edition. This book was so much easier to write 10 years ago. Computers were relatively new, and they were all pretty much the same. There were only a few important differences, whether it was an Apple or a Wintel, that is, running a Windows on an Intel processor. There were some variations in size, portability, and customization. Enthusiasts could do by installing different drives and expansion boards inside PCs. Stuffing the newest fangled components to my computer at PC Computing, the magazine where I worked, was a wonderful pastime and the source of many stories in the magazine. If I made a mistake and the computer groaned its last breath, that often made for an even better story. One reason PC disasters made for good reading is that every computer user had a similar story of computer disaster. We were all in the same boat because even the editors of the computer magazines, if truth be told, didn't know what they were doing a lot of the time. Back then, if you took a class in computers in college, you waited your turn to run a program on some monster of a computer that would return a solution in the form of punch cards. There was no formal place to go to learn about PCs. Manuals were useless beyond finding out how to plug a PC in the wall and where the switch is located. Programs that worked one way had no resemblance to how other programs worked. You learned about computers by trial and error, by going to user groups made up of equally befuddled PC owners and by reading magazines. Before I went to work for PC Computing in 1989, my teachers were PC Magazine, PC World, Byte, Popular Computing, and other magazines covering this new technical phenomenon. You found a morsel of information in one magazine and a nugget in another, and eventually you pieced together what was going on inside that steel box on your desktop. But I was going to tell you why it was easier to write this book 20 years ago. Back then you could say all someone needed to know about a floppy disk and a couple of 2-page spreads. Something as mind-busting and as intricate as a monitor you could do with one illustration. The technology was in retrospect elementary. How computers and their components worked was actually simpler when you sketched it all out and added some callouts and arrows. No wonder I figured I could, with the help of a great illustrator, write a book that laid out all the mysteries of what was really a fascinating technology. But today, Lord love a duck. Today, there's not even a simple explanation of what a computer is. Simply having a microchip doesn't cut it. Microchips are everywhere in watches, cars, thermostats, ovens, refrigerators, flashlights, dog collars, coffee pots, even cats. Just about anything that uses electricity. But these aren't computers. They're just electronic doodads that use the abilities of microprocessors to perform routine chores. To be a computer, something must be programmable. It must be capable of doing different things based on the instructions you give it. Your yard sprinkler system is a computer. True, it doesn't do a lot of different things, but you can program it to water different parts of your yard for different lengths of time and on different days, or not at all if it's raining. Microwave ovens are computers you use by programming it on the fly, by setting how long it will cook at different temperatures, or you can push a popcorn button to tell the oven to use a pre-programmed sequence. These are all computers, but the truth is they're not very interesting. How deeply do you care about how your sprinkler works? But a computer that can be programmed to do so many different things, now that's a computer worthy of the name. And we see them everywhere today. Phones, cameras, tablets, music players, and TVs have all become computers that are indispensable parts of everyday life. Our lives are richer for the knowledge, problem-solving, communication, entertainment, health, and income they provide. They certainly deserve to be included in a book that calls itself How Computers Work. The problem, my problem anyway, is that they make organizing a book like this an exercise in controlled chaos. In previous editions, I simply had a section on storage, another on displays, and one on networking. I admit that in this edition I've fallen back on that system in some chapters out of pure convenience, but certainly the workings of today's computers are not so compartmentalized. The more I looked at computing in the last 20 years, the more it struck me that the real story of how computers work is how they are like living creatures, organisms that have evolved and continue to evolve. These metal and silicon and plastic and glass animals have followed the same patterns of mutation, natural selection, and survival of the fittest that Darwin found in plants and animals. I've tried to explain how technological developments following the same rise and fall that carry organic species to new shores. Today's achievements in computing are possible only because they rise out of the fossil beds of the earlier technology. There is no way for today's technology to have been invented from scratch. I recall that some 30 years ago I was working for a computing service company that relied on mainframes, an obsolete term to describe computers the size of a couple of high-end refrigerators slammed together. They used two kinds of storage, hard drives and magnetic tape. The 5 megabyte hard drives were the size of large garbage cans, and the platters where information was stored could have doubled as hubcaps. You'd had to have seen the magnetic tape at work to understand one of the computer's most common terms, RAM, random access memory. It sounds as if data retrieved willy-nilly from all parts of the memory chips with no order to it. Memory chips got their RAM moniker because retrieving tape memory was not random. If you wanted data stored on a reel at the innermost 10 inches of tape, the computer had to wind through the first 3,500 feet to get that data. Sure, it would have been better to skip tape altogether and go straight to hard disks, but to paraphrase a certain infamous quote, sometimes you don't build computers with the technology you wish you had, you build them with the technology you have. Life in the primordial seas had to get the hang of existence as one-celled organisms before the cells could master hooking together and dividing up the jobs needed for mutual survival. Some of the bigger organisms learned to move by wiggling. They developed a way to filter oxygen from water, and later that system developed into lungs that could suck oxygen out of water. It took millions of years for these changes to evolve. You couldn't create magnetic disks until you learned how to record data to magnetic tape. Then it took years of engineers trying first this and then that to make the disks smaller and at the same time more capacious. Current solid-state drives come from the fossils of technological forays such as bubble memory that didn't survive. Maybe we'll talk about core memory some other time. Core magnetic memory is very interesting. Old first memory made by DEC. The most amazing aspect of this whirlwind of technological progress is that it's only taken 20 years to go from floppy to solid state, from displays that amounted to stick figures to animated graphics that are almost indistinguishable from film images. That smartphone in your pocket is more computing power than those refrigerator-sized mainframes. The wires that have been needed for communications for a century are likely to disappear completely in the next 10 years, along with the need for local data storage. It's possible the keyboard and mouse will become extinct. In fact, much of the hardware we're used to today, as it gets smaller and more efficient, may migrate from our desks and our pockets to become parts of our eyes, ears, and brain. And that's just in the next 20 years. Beyond that, we can't imagine what will become of technology any more than single-celled organisms could have imagined a dinosaur. Let's check back with each other after a couple of decades and then see what may be possible. All right, this is a battery and a light switch. Chapter 1, what makes a computer a computer? If you want to find the secrets of the universe, think in terms of energy, frequency, and vibration. Nikola Tesla. Those of us who studied physics in high school half a century ago had to learn about the 6 simple machines. There was the wheel and axle, the lever, the pulley, the inclined plane, the wedge, and the screw. The explanations of how they saved us labor were invariably accompanied by illustrations of people who looked like they were working awfully hard. Just as invariably, someone would quote Archimedes saying, "Give me a long enough lever and a place to stand, and I will move the earth." Note that Archimedes gave somebody else the really hard job, finding a lever that would have to stretch from Earth to Jupiter. Still, in the days before any form of power, other than the muscles of man and horse, the simple machines were ingenious. Those 6 machines were combined in various ways to create wagons, catapults, irrigation systems, roads, bridges, and, need I mention, the pyramids. Put some teeth on a wheel and you get a gear. Combine gears with levers and pulleys and you have a clock. The ingenuity with which clever people combined simple machines into complex and powerful machines grew every century, abetted by engines running on steam, water, compressed air, and chemical fuels that magnified the power of these simple machines, turned complex to a point that even Archimedes didn't imagine. Then came electricity. Here was a mysterious force that would run through the right kind of metal wires. Those wires could be bent and wrapped around other wires and in pieces of metal to produce an engine that, unlike water mills, was portable. One that, unlike steam and chemically fueled engines, didn't threaten to blow up. If electricity had produced no more than a safer, more efficient motor to run all these complex and simple machines, that in itself would have been a boon to civilization. But scientists and engineers discovered new properties in this force, despite the fact they didn't know what it was and couldn't see it. You know the results, everything from the electric light bulb to the computer, smartphone, tablet, in which you might be reading this very book. Electricity really began to pay off when engineers looked beyond its ability to make a filament glow or a motor turn. They began to discover more possibilities in electricity's partnership with magnetism, in the electromagnetic fields produced by electricity that would interact with other matter, even over great distances. They found that electricity and light were the 2 facets of the same thing, an electromagnetic spectrum with mostly invisible fields made of waves vibrating at different frequencies and the sizes which range from the microscopic to the earth encompassing. Like the simple machines of medieval ages, these new elementary principles could be combined in endless ways to create new tools to accomplish jobs that were previously unimaginable. The real power of electromagnetism, engineers found, was less in its ability to move trains, see inside a patient's body, and turn night into day, than it was to store, manipulate, and distribute information. But all these uses are some very fundamental principles. Behind all these uses are some very fundamental principles of physics that have led us to the brink of discovering the true nature of everything in the universe. It's these simple principles of a new age that you're going to look at in the first part of this book. I can already hear some of you saying, hey, when do we get to the stuff about computers? You're there. The subject of these first chapters are the stuff of the computers. And it doesn't matter what type of computer you're talking about. Mainframe, desktop, laptop, table, digital music player, camera, GPS, smartphone, smartwatch, or glass. All are computers, and all are work using some version of the principles and technology you'll find here. Chapter 1, the ghostly Legos of computing. The entire world right now is run by information. Our entire world is being controlled and operated by tiny invisible ones and zeros that are flashing through the air and flashing through the wires around us. Chris Doyen, computer activist, hiding out in Canada. Louis C.K. has a routine where his daughter asks a simple question that leads to a metaphysical issue people have pondered for ages. It begins with a question about why she can't go outside in the rain, and why it's raining, and why are there clouds, and it becomes a dissertation on why Louis doesn't know more, and why he didn't listen in school, etc. The string of answers finally lead father and daughter to an ultimate question. Because some things are and some things are not. Why? Because things that are not can't be. Why? Because nothing would be. Understanding how a computer works is a lot like that. You say a hard drive uses a magnet to write files, but then you're obligated to explain why. And then you have to say why magnets attract certain metals, and then explain why magnets have these invisible force fields. And don't expect your computer to explain itself. It maintains a sphinx-like silence. Shouldn't there be doodads and thingamajigs that are pumping or swinging, flashing or beeping, vibrating or rotating, or at least ticking and talking? For all you can tell about your computer works from opening it up and looking inside, your computer might as well be carved out of soap. In your computer, the things that make it really work are all ghosts. You've never seen them. You see only what they do, like a lamp thrown to the floor by a poltergeist. And computers, from PCs to a Galaxy X phone, tightly tuck away their secrets. You have to keep asking why. And if you ask long enough, you get into physical states filled with objects so small you can't see them, or that exist only in ghostly states, such as the vast parts of the electromagnetic spectrum or some screwy spinning quantum string, all of which are invisible to mere humans. The only way to get even a hint about what these little ghosts are like are to fling them around in a giant mile-wide magnets, like the Large Hadron Collider, until they smash into each other and create even smaller, rarer ghosts that vanish as soon as they appear. We're just left with only the contrails they leave behind, and from those we try to figure out what just happened. These little invisible ghosts are the ultimate things, the tiny invisible ones and zeros, as Doyen says. The ghostly little digital Legos that unite in different combinations to create everything in your computer, tablet, or smartphone. To understand, really understand how a microprocessor, a monitor, or a printer works, you have to understand the ghostly workings of electricity, waves, magnetism, binary math, and data packets. So let's examine them all. Don't be frightened. They're friendly ghosts, I think. How waves take the universe on a joyride. Waves do far more than carry surfers on a wet thrill ride. Waves are essential to the working of computers. They move data from one place to another and provide the beat that every PC component marches to. Without waves, we'd never be able to see the solutions, images, or words that result from the machine's computations. Without waves, we'd be beached, or we might not exist at all. According to some theories of physics, waves are the ultimate stuff of the universe. Space, Earth, you and I are no more than the interference patterns created by the crisscrossing of waves, which are created by tiny, tiny, tiny vibrating strings. Indeed, at times it seems as if the thing we think of as solid Earth is just an illusion. Under the right circumstances, waves pass through our buildings, rocks, waters, and other waves, and even our own bodies. It's as if the Earth is no more than a wad of soppy tissue. Here's how this humming master universe reaches down to play its tunes on the computer. If you've ever played with toys, you're familiar with the two most common mechanical waves in the universe, transverse waves and longitudinal waves. When you and a friend move the ends of a jump rope up and down, the graceful humps traveling the rope are a transverse wave. Here's a transverse wave. Trough, crest, trough, crest. Here's the amplitude, and the wavelength. There's a different distance between two troughs. Examples of transverse waves include light, surface earthquake waves, surface water waves, magnetic waves, and the wave people in a football stadium create. Longitudinal waves are found in the depths of water and earth. A tsunami or earthquake moves tremendous energy through these media until they reach the Earth's surface or the shallows of the sea, where they convert to transverse waves. Freeway traffic jams, with their alternative congestion and fleeting moments of faster driving, are also longitudinal waves. Here's longitudinal waves with compression, dilation, there's a wavelength, there's an amplitude, and an undisturbed medium that will soon be disturbed. Both types of waves can carry energy, information, and destruction. They can be measures of time and distance. Both types are created when some force, like vocal cords, a rock thrown in water, a meteor smashing into Earth, is exerted against their mediums. Transverse waves can travel through solids and liquids, but they do not travel through gases because air does not have the elasticity needed to push the wave's vibrating molecules back into place. Only longitudinal waves may use gases as a medium. Despite that difference, longitudinal waves can morph into transverse waves and vice versa when they confront a different kind of medium. Both waves have similar components. In both types of waves, two fundamental measurements are the wavelength and the frequency. Wavelength is the distance between one crest or compressed area to the next. Frequency is the number of waves measured by wavelength that pass through a stationary point in 1 second. The longer the wavelength, the lower the frequency. Frequency is measured by hertz. Generally, the higher the hertz, the more energy a wave contains. In audio, hertz means a higher pitched sound. So hertz is the number of cycles of a second. Transverse waves in the water and longitudinal waves in the air are both called mechanical because they depend on the mechanical mechanism of their media to propagate. That, however, is an illusion. The energy that goes into creating and maintaining waves is transferred by molecules knocking against each other, similar to how runners in a relay race hand off the baton. Next time you're at a lake or beach, observe how a bird or boat floating on the surface of the water only bobs up and down in place when a wave passes under it. Its horizontal position does not change. Another type of wave, electromagnetic, is not mechanical. Electromagnetic waves encompass light, infrared light, radio, X-rays, gamma rays, and neutrinos. They're all part of the electromagnetic spectrum in which waves range from a wavelength smaller than an atom to wavelengths bigger than the Earth. Take a look at this. Gamma rays are 10 to the negative 12th. This is the radiation length. X-rays are shorter. Ultraviolet is nearly visible. Then we have visible light. Then we've got infrared light, microwave light, and radio. Radio is very long wavelengths. The scale of the wavelength is interesting as well. There's the frequency. The higher the frequency, the lower the wavelength. I hope that makes sense. And this is the temperature of objects at which this radiation is the most intense wavelength emitted. Interesting. Electromagnetic waves do not require any type of media and are the only waves that can travel through the vacuum of space. Although not mechanical, electromagnetic waves are transverse, doubly transverse. They consist of electric and magnetic fields that, like other transverse waves, vibrate perpendicular to the direction in which the wave is moving. But the two fields vibrate at right angles to each other, and each expansion and collapse of a field creates an opposite field. The vibrations of electrical fields generate magnetic fields. Vibrations of magnetic fields generate electrical fields. The result is a never-ending bootstrap that pushes the wave forward at the speed of light. How information rides the waves. What do surfing, lasers, computer circuits, televisions, rock concerts, and the ever-popular death ray have in common? They all depend on waves. Without all the types of waves that surround us and that penetrate us at all times, and without the ability of waves to carry information nearly instantly to all parts of the globe, chances are civilizations we bask in it today would have never have happened. It takes no great technology to use waves to send information. You simply turn a wave on and off. It's the basis for jungle drums, Morse code, the earliest modems, and it's called encoding. Regardless of the form of encoding, the sound or electrical waves are not changed. Information is encoded in how often a wave is created and how long the wave exists. Morse code and the first 300 baud modems use short and long pulses of electricity to create a simple binary code, a forerunner of how data is stored and transmitted to computers. The short dits and the long das in different combinations represent different letters of the alphabet. A total of 9 pulses of electricity are enough to code the 3 letters that represent SOS, the Morse code distress cry. Note that the frequency and amplitude of the electrical signal does not change. only how long the same electromagnetic signal is broadcast. Here's a high level of noise when the key is pressed, dit, 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 da, 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 dit, dit, dit. Little or no signal when the key is not pressed. Long and short drum beats are not possible with jungle drums. They depend on recognizable patterns in which volume of individual beats and this length of silences between the beats create a pattern that can convey information. Because we're not fluent in jungle drums, here's the visualization of a 22-second drum solo by Gene Krupa. The breaks in the waveform depict bass drum beats. At the right end of the segment, you can see how intervals between the bass grow shorter and shorter, creating the type of pattern we could, if we didn't have all our phones coming out of our ears, use for communications. Amplitude modulation makes changes to the amplitude of a wave. Frequency makes change to the frequency. Jungle drums and Morse code have obvious limitations. Both are slow and depend on a skilled person doing a hands-on job. What is needed are entirely different modulation methods. 3 modulations methods do the job. FM, frequency modulation, AM, amplitude modulation, and pulse modulation. Pulse modulation is the newest way to add information to waves and get information out of them. It was developed because modems had reached a limit as to how quickly and accurately they could turn a signal on and off. The solution was to use different frequencies or amplitudes to stand for combinations of letters. This scheme is an enormous improvement over early modems, but is limited to text and digital information. Transmitting analog signals without first converting them to digital signals requires AM or FM. Either AM or FM can be used to convey information over radio waves or electrical circuits, using frequencies from 100,000 hertz radio through microwave to 10,000 billion hertz infrared. The higher the frequency, the smaller the wavelength. Radio waves are about as long as your average skyscraper is tall. Microwaves are the length of an adult person, and infrared waves are the size of a computer mouse. Various factors govern a wavelength's usefulness for carrying data. Some radio waves penetrate brick and steel buildings. Others bounce off of them. Generally, the higher the frequency, the more data it can carry, because the more often a wave moves between peaks and valleys, the more opportunities there are for modulating it to carry information. Interesting. You always wonder how the radio works, right? To understand modulation, go to a pond or a lake and throw a rock in the water. If you throw only a single rock, it creates waves that expand in concentric circles, smoothly and evenly from the point of impact. We call this our carrier wave, and measure its wavelength, how quickly it expands, and how tall and deep its crest and trough. As the carrier wave expands, toss a few pebbles across the width of the carrier. The pebbles make waves of their own. The pebbles' waves change, as in modulate, the carrier wave whenever they meet. If we measured all the directions the combined waves move, their speed, and where they eventually meet the shore, and then, using some sort of math I don't even want to think about, we subtracted the influence of the carrier wave, we'd be left with the information we need to know, where each of the pebbles struck the water and the force behind each. A radio carrier signal, like the watery carrier wave, is a simple steady wave at a specific known wavelength, like 92.3 here in New York. We mix the carrier wave with a wave known as an input signal created by the sound or sights from another source. The two waves combine into a modulated signal that is then broadcast over the airwaves or through wires. When a radio, TV, or other device receives the modulated signal, it removes the influence of the original carrier wave. What remains is a replica of the input signal that created the modulation. How electromagnetism is the ghost in the machine. Just got a couple pages left, and then we can turn to something maybe a little more sophisticated, but I do like this. It's a lot easier to understand than the book on shift registers. How Electromagnetism is the Ghost in the Machine. This book begins with a quote from Arthur C. Clarke: "Any sufficiently advanced technology is indistinguishable from magic." Well, we're still not past the first chapter, and it's already time to start the magic show. Like good magicians, electricity and magnetism, the twin powers of electromagnetism, hide what they're doing. They mostly show the results, the rabbit coming out of the hat, not how it got into the hat. We can see the effects of magnetism on iron filings, refrigerator doors, and the giant generators that supply much of our energy, but we cannot see magnetism itself. It's a silent partner in the electromagnetism spectrum. We can see electricity in the form of lightning and sparks from a shorting wall plug, and we can certainly feel electricity when static electricity jumps from our fingers on a cold day. Static electricity is a buildup of surplus electrons. Combined, magnetism and electricity form the electromagnetic spectrum, the full frequency range of the mysterious waves that permeate the universe. This partnership continues into other areas where these conjoined energy ghosts form the basis for the most modern of technology. for most of modern technology. Electricity and magnetism cannot survive without each other. The previous illustration shows that magnetism and electric fields cooperate in a bootstrapping operation that allows electromagnetic waves such as light, radio, and heat to travel through a vacuum. Let's start with the electro half of electromagnetism. In some atoms, such as copper and silver, the attraction is weak between the atom's core, a nucleus made of positively charged protons, and the negatively charged electron clouds in the atom's outer layers. In such conductive materials, it's possible for electrons to jump freely from one atom to another. This movement of electrons, which occurs only under the right circumstances, is electricity, and it travels at the same speed as light, another form of electromagnetism, which is 186,000 miles per second. All right, we apply 3 measurements when working with electricity: voltage, amperage, and resistance. It's convenient to think of voltage, or charge, as being like water pressure, how much power is behind electricity. Amperage, or amps, is the measure of how much electricity is flowing through the wires, similar to how much we measure gallons of waters that pass through pipes. And resistance is how the material through which the electricity is flowing. In these examples here, higher water pressure overcomes a clogged pipe, and doubling the voltage pushes the same amount of current despite the addition of a resistor. So here's battery, the resistor I mentioned a while back on the cover. Both readings the same on an amp meter measuring voltage. In some materials, such as rubber and glass, electrons are more closely bound to their nuclei and do not easily move from one atom to another. These non-conductive materials are called insulators. Still, other materials like silicon can act as either conductors or insulators depending on the conditions. These are semiconductors, which are an important component of microchips and transistors. Lower pressure, less resistance with a clear pipe. Same amount of water gets through when you have higher pressure, and more resistance when the pipe is clogged. When electricity moves through a wire, it generates a magnetic field that surrounds the wire. The magnetic field is a straight piece of wire is too weak to pick up even the smallest iron filings. Conversely, when a wire moves through a magnetic field, the interaction creates an electric current in the wire. Note that in order for either of these phenomena to work, the wire must be moving through a magnetic field, or the magnetic field needs to be collapsing or expanding across the wire. Both magnetic fields caused by electricity moving through in a wire and electricity created from magnetism can be made more powerful if the wires are wrapped around iron cores. Almost all electricity we use comes from generators, in which massive magnets surround equally massive coils of wire. Water from a dam or steam created by a nuclear plant or fuel-burning plant spins the wire coil rapidly, creating a high-voltage alternating current that is transmitted via thick, low-resistance cables to electrical substations throughout urban areas. Interesting. How do we control electricity? Straight from the generators, electricity measures in the thousands of volts, much more powerful than we need for everyday uses. Even in the home, the voltage of current from a wall plug is many more times more than most household electronics need or can tolerate. Within those same electronics, different components require still smaller and varying voltages and amperages, higher or lower than the current flowing through most of the circuitry. How do we control all this? At a power station, steam generated from burning coal or heat from a nuclear plant turns a shaft within an electric dynamo. The shaft is attached to a rotor covered with thousands of wires called bushings. The rotor turns inside 2 permanent stator magnets whose fields generate high voltage electricity in the bushings that is transmitted as alternating current to the public power grid to homes and businesses. There's the stator magnet, the rotor, and the current comes out, I guess. There's the residential power transformer. Before the electricity reaches its final destinations, it passes through the transformer to reduce the voltage. The transformer has a common magnetic core, usually iron, around which 2 wires are wrapped. Alternating current, electricity that flows first in one direction and then another, moves through the primary coil, the wire that has more wrappings around the core. The transformer cannot use direct current, which flows only in one direction because the magnetic field cannot create electricity in wires unless either the wires or the field are moving. Interesting. Each time the current switches directions, its magnetic field expands or collapses, which is the same as the field moving, and induces electric current in the coil. The current passes through the part of the core which is wrapped with the secondary coil. There the AC produces another magnetic field and it envelops the wire wrapped around the core. The field creates a current in the secondary coil, but because the secondary has fewer wrappings, the current created in the secondary coil has a smaller voltage, making this a step-down transformer. A step-down transformer, which is most likely mounted on a utility pole near your home, cuts the voltage of the raw generator current from thousands of volts to 120 volts before it's allowed inside. Huh. There's a step-up transformer and there's a step-down transformer. Before a computer can use the household current, the current passes through a second step-down transformer, this time in the PC's power supply. The box festooned with warnings not to open it, that's at the back of the computer. This transformer reduces the 120 volts current into two different voltages that separate wires carry to 5 volt and 3 volt components. Other electronic devices, such as amplifiers, use a step-up transformer to raise the same 120 volts to a level powerful enough to drive speakers. We use resistors to decrease the flow of electricity, much like we use a valve to limit how much water flows through a pipe. Resistors are commonly made of carbon, plastic doped with metals, and metals such as nickel chrome that do not conduct electricity well. Varying the materials and their size and shape changes how much resistance the component has, measured in ohms. Color coding tells how many ohms of resistance the part provides and the percentage by which the value may vary. I used to know how to read these. A spiral cut in the resistance material determines the resistance value. A narrower spiral increases the ohm value of the resistor because it creates a longer path the electricity must follow. Does anybody know the color coding of the resistors? Variable resistors, such as the rheostat, shown here, it's a great term, are used in situations that call for the resistance to be changed manually on the fly. The best example of a traditional dial controlling volume on a radio. The outer ring, or toroid coil, consists of a resistive wire wrapped in a spiral about a ring of insulating material. Cool. Maybe you'd have to make this yourself in a desert island, or a deserted island. Do you think you could do it? Electricity must travel along a sliding wiper and through the coil before it leaves the resistor. When the knob is turned clockwise, electricity has less wire to travel through so that the resulting current is stronger and the volume is louder. Capacitors, oh man, these are old friends of mine, are made of two conductive materials that sandwich a material with limited conductivity, such as plastic, ceramic, or even paper. When current is applied to the two conductive terminals, the electricity does not flow through the capacitor. Instead, electrons from the power source pile up at one end of the terminals called the anode, like logs swept on a dam. They leave a dearth of electrons on the second terminal, the cathode, so that it is charged positively by the protons that have been deserted by the electrons. The charges remain in the capacitor for a few seconds to several minutes, depending on the design. When a circuit connecting the two terminals closes, the capacitor discharges, the electrons jump the insulator, to send a bigger than usual burst of current needed for, say, a photoflash or something like that. Larger capacitors, such as those found in CRT monitors, cathode ray tube monitors, and some power supplies can be dangerous or fatal if handled improperly. So there's the kind of capacity building. How computers create our world using numbers. So this is sort of ties it all together. Waves, electromagnetic fields, particles, electrons, light waves, and quarks. These are the elements by which computers operate or compute, measuring, comparing, adding, and subtracting. All that computing brilliance has to be communicated to us less brilliant organic units in some way. Unfortunately, the native tongue of computers is numbers, which most of us stop thinking about within days of completing Algebra 2. Because we're not fluent enough in numbers to enter the world of computers, computers have devised ways of using numbers to recreate our world as we understand it. Their numerical constructs are so smooth that we're rarely aware that they are made of only ones and zeros. There are two ways to take stock of the world around us, analog and digital. An analog instrument expresses measurements in a manner that is analogous to whatever it's measuring. An old-fashioned thermometer is an analog device. It has a glass tube filled with red-dyed alcohol. As the thermometer gets warmer, the heat causes the alcohol to expand and rise within the tube. We can look at the markings along the tube and say that a person's temperature is about 99.5. We say about because a reading of the temperature depends on the precision used in making the thermometer and the acuity of our vision. A digital thermometer has an electrical component called a thermistor. Rising temperatures make the thermistor less resistant to electrical current, allowing an increased electrical flow to a mini processor that converts the amount of electricity to numbers that represent the temperature. The numbers are displayed on a screen, but unlike the alcohol in the analog thermometer, the numbers don't swell or shrink in proportion to temperature variations. Most of today's computers are digital because the transistors that jumble data are digital, either on or off, and not some state in between. Analog computers are used in situations involving constantly changing measurements, such as the automatic transmission or a bombardier's sight. Digital computers, however, often start off with analog measurements that must be converted to digital. A computer-based digital camera replaces film with an image sensor, a microchip studded with photodiodes, which are transistors melded with substances that convert photo energy into electrical energy. Each of the photodiodes collects photons of light, building up electrical charges as long as the shutter is open. The brighter a part of a scene, the more photons are focused on pixels capturing that part of the scene. When the shutter closes, all of the pixels have electrical charges that are proportional to the number of photons that hit each photodiode. If you picture photons piling up in mounds of glowing pebbles in buckets, some larger or smaller, you have the idea. The camera circuitry moves the charges collected by the image sensor into a single row that passes through an amplifier. The amp converts what are little more than faint static charges into an electrical current with varying voltages in proportion to the parade of changing charges. The current passes into an analog to digital converter, ADC. Every few milliseconds, the ADC measures the voltage of the current as it passes through. If the voltage is above a certain level, the ADC produces a 1-bit, which it adds to a stream of digital values that it passes on to other components. If it's beneath a certain level, the ADC adds a 0-bit. Data that has only two values, 0 and 1, doesn't have the versatility to represent in an analog format the real world of images and sounds that, ironically, have become the most popular use of computers. With only two values, such as black and white, and no grays, an image looks like this 2-bit Mona Lisa. One can obtain more precise digital representations of analog signals through multi-bit sampling. The current being measured, whether generated by energy of light, sound, or chemical reactions, passes through a series of resistors. Each resistor depletes some of the current until it's no longer measurable. How many resistors it passes through determines a value, depending on the design that is represented by 2 bits, 4, or 8 bits, the bit rate. Diagram A on the far right, with a bit rate of 4, there are 4 values the voltage could reach for each sample. With diagram B on the right shows an ADC capable of registering 8 bits per reading. Diagram B also demonstrates how the conversion's resolution is made more accurate by increasing the sampling rate, the frequency at which samples of passing current are measured. In diagram A, 8 samples are taken for the period shown. Diagram B shows a sampling rate 4 times greater. If diagram A were representing the digitization of a music recording, its sampling or frequency rate might be appropriate for a smooth slide on a trombone. Diagram B has the same general shape as the red line in A, but the higher sampling rate provides opportunities to capture variations in sound that the first can't handle, for example, a rapid trill on a clarinet. Having 16 bits for each sample might not sound particularly useful, but 16 bits in a binary number yields 256 different combinations. In creating video color, that means 16 bits can produce any 256 shades of a color, as shown here with red. A bit value of all these zeros is this 16 zeros. Binary, or 0 and decimal, is assigned to total black. All ones, binary or 256 decimal, represents a pure and intense a red as a monitor can produce. Take any 2 adjacent red squares shown here, and the difference between them is less than the human eye can distinguish. Combine 256 shades of red with 256 shades of the other 2 colors that go into a video image, green and blue, and the result is an amazing 16 million possible colors. An ADC is a functional mirror image in a DAC, or a digital to analog converter. Some monitors and most speakers require analog currents. The DAC changes a string of digital values into rapidly changing voltages in electrical current by routing a feeder current through a matrix of resistors that partially block the current. The resistors are weighted to represent different degrees of resistance. Sending current through resistors on different pathways turns out a final stream of current that corresponds to the digital data. That's how our speakers work. And that's chapter 1 of what makes a computer, or how does a computer work? How computers work, which is CS50 suggested reading in Harvard. I hope you enjoyed that. We've read 3 of these now for, let's see, I guess 1.5 Harvard courses. We have a few more options. This is a C program from before. That prints out itself, I guess, which is kind of neat. There's general chemistry, there's linear algebra, there's statistics, and this is the biology that we were getting through to. We ended here on one protozoan eats another. This is a scanning electron micrograph that shows didinium on its own with its circumferential rings of being cilia and its snout at the top. And here it's eating a paramecium, which is colored yellow. Pretty wild. Let's check in on our cat. The cat is asleep. I have to get the cat some tuna. I think would be a good idea. Analog computing is very interesting concept. I think you accrue knowledge as much as you can, and then over time, you know, you can use it when you need to. There's no rush. I have so many name suggestions. Some of these are really good. Little scrambles. She's dead asleep. Poor baby. I don't think SBF is going to get life, but he may have to do some time. Yeah, I don't think retention is a good idea. Let's see. There's, no, I think SBF will get longer than 5 years. I think he'll get like 20-something years. All right, so we have done a few of these books. We've got biology, statistics, chemistry, linear algebra left. And then I probably want to get a physics book. And what else? Should we do something from the humanities? SBF's bail is very restrictive. Right now he's on basically house arrest. Linguistics and Noam Chomsky. Maybe we should just look at whatever they offer. Maybe some, I mean, I'd pick linear algebra for math. So let's see, should I get some tuna for the cat? What do you think? I think she wants some fish, tuna fish. We'll have our first tuna party. What do you think? Tuna party? She's asleep and she doesn't even know that we're planning a tuna party. Can you see her? I don't know if you guys can see her. She's slumped over there. Oh, okay. Churu. Oh, cats love churu. Should I zoom in? Think I can zoom in? Let's see here. You want just cat? No Martin? Beta? Maybe. Would you guys give me 5 minutes to go get some tuna? Get this tuna party started? She's not a sphinx. I would never have a sphinx cat. Okay, so I'm going to get paper plates, tuna. I need a scooper. You guys just take care of her while I'm gone, okay? If anything happens, just shoot me a text, all right? I don't know if this is a good idea. What's up, Mochi? What's up, Mochi? Purr. Purr your little heart out. Purr. Yes, I love you too. Yes, I love you too. Yes, I do. Yes, I do. Why you purr so much, huh? You want to meet your older sister, Trashy? Yes, I love you. When am I going to feed you? I'll feed you soon. Yes. Oh, you're a little baby, huh? You're a little baby. Now you sit there until I get back. Don't you move. Snoopy? Oh, hey! Why are you hiding? Oh, hey, yeah, it's me! Yeah, what's up? We got a tuna party. What kind of meow is that? Give me a big roar. Come on. That isn't a meow. Meow. That's not a meow. What kind of meow is that? That's not even a meow. Guess what? We got tuna! Oh my god! Oh my god! Tuna! Tuna party! Yeah! Tuna party! Wow! I know! You excited? Yeah! Yeah, I know! I love you too! I love you too! Tuna party, here we go. Tuna party, meow meow meow. Oh yeah, you know what's coming. You know what's coming. You know we're gonna have a tuna party. Are you ready? Oh my gosh, you can smell it. Oh, you can smell it. It smells so stinky. No, hold on, I gotta chop it up. No, hold on. I gotta chop it up. Hold on. Hold on, I gotta chop it. All right, Your Majesty may begin to feed. Enjoy. It's a tuna party. Yeah, it tastes good. You don't want it? So help me God if you don't eat this. I'll turn you to kitty stew. She does this, it's weird. Just before she eats, she likes to scratch a little. What are you doing, kitty? Eat it. Yeah, you hungry? It's delicious. Here you go. Stupid cat. Eat that, kitty. Oh, come on, kitty. Eat your food. Stupid. If you don't eat that tuna,
We're having a tuna party. You barely touched it. You're a small kitty. You gotta grow. You gotta grow to be a ferocious beast. What's that? What kind of meow is that? What? You're 2 pounds. Eat up, kitty. Yeah, I love you too. Now eat. Stupid. Here, come here. This food is so yummy. Oh, yeah. That's why you're the runt of the litter, huh? That's why you're the runt. You're mangy, good for nothing kitty. You're about 2 pounds soaking wet. Okay, yeah, take a little, you're just smelling it. What are you scraping? You're not going potty, you gotta eat. You get your signals mixed up in your brain. What, now you stepped in it, stupid. This isn't litter, this is food. You're all scrambled up, kitty. No. You got your brain scrambled. No, you're not going potty. I gotta take you back, you're defective. I think you're a defective kitty. What are you scraping? Just eat. Dummy. I feel like I'm watching National Geographic, the unusual Shkreli cat. Instead of eating, there we go. Stupid. There we go. Good girl. Yeah, keep eating. That's not enough. You're gonna eat more. Okay, now why are you doing that? I'm gonna make kitty stew out of you. You're done. Where's the ruler? I'm gonna give you some kitty discipline. Give me the ruler. Where's the ruler? Yeah. Yeah, the ruler's coming. Because you're being a bad kitty. It gets the ruler. I'm just kidding. Gonna hit you with the ruler, kitty, if you don't eat. Now what are you doing? That's not the wrong thing. That's for poo-poo. You do that after. You're a little dummy. I need to take you back to the kitty store. Or just make some kitty stew out of you. And everyone's gonna ask, what happened to that kitty? And I'm gonna say, I ate him. I ate her. There we go. No, don't. Yeah, just eat it. No, stupid. This kitty's broken. No, you're not supposed to do that. You're supposed to eat it. Is that why you're the runt? You don't know the difference? Hey, this is delicious pate. Ocean fish, yeah. No, just eat it. No. You just eat it. No, you don't have to do that. What are you doing? You're broken. No, you could just eat. No. This is for you. Not maybe because she was the runt of the litter. The hell kind of cat is this?
gonna rewire you, kitty. You're gonna get rewired. I'm gonna have to perform some surgery. Do you want a little surgery, kitty? You need a little... Yeah? You need a little surgery? A little lobotomy? Yeah. Yeah. Yeah, I know. I know. Yeah, eat some food. Some food, Mama. Come on, Mama. Got some fish. Alright, you know what? I thought you would be difficult. So what I did is I got you these. 3 mouse toys. That's right. Fuzzy mouse cat toy. Thought you'd be a little... No, that's different. Yeah, eat it. Why? What is this Pavlovian mess? She even puts her foot in it. This carpet's gonna need a good dry cleaning. Are you hungry? Are you... This is why she's a runt. No, you're supposed to eat it, not... Yeah, eat, good. No! She got everything backwards. Maybe I do have this. Maybe I have everything on backwards. Send me a link on what? Confused cat? She's taking too much tirzepatide? I knew I would just have to run experiments on this cat. Yeah, I am going to run experiments on you. I thought I'd get a cat that I could hold and love and nourish and take care of and nurture and be my companion. But you suck. You get an F as a cat. Yeah, that's right. You get an F. You get an F. You get an F, and you're going straight to the garbage, and you'll be somebody else's trashy cat. Yes, you will. Hey, look at that! Look at that kick! The karate kick. Okay, sorry. I won't hug you again. What's this though? Is this a little mouse? I believe it is. I believe it's a little mouse. The mouse don't like you either, by the way. The mouse don't like you either. We need kitty cam. You want to fight him? Oh, you want to fight him, huh? Woo! Yeah, beat him up, beat him up. Give him the 1-2. Simple toy. How could I possibly be entertained by this? I'm merely calculating eigenvalues. I'm not playing with this. Oh, you sneezed. God bless you. God bless you. You think she's the Queen of England? Now let me move this computer out of the way. Let me get this out of the way, kitty. Are you a bad kitty? Tell the truth. Yes, you are. You're a very bad kitty. Admit it. Admit it already. Plead guilty to 5 charges of being a bad kitty. We'll go easy on you. Oh, you want to be a tough kitty. Okay. will supersede you with a 20 count indictment for bad kitty behavior. Okay, okay, okay. Take him away. Take her away. Is this what you're going to do to a putative mouse invader? Kitty, if we weren't on webcast, I'd be whipping you with the ruler. Whap! Whap! It's what you deserve. All those people at the cat adoption centers who denied me because I'm a felon, they made the right call, Kitty. They made the right call. Because you and me, we're loners, Kitty. All we got is each other. You got litter all over you. You got litter all over you, stupid. Yes, I love you, I love you. I know you've been a ferocious beast attacking this felt toy. Kitty, you're a little dum-dum, huh? You're a little dum-dum. You're a little dum-dum, eh? Ow, no, do not scratch me. You do not scratch me. Ever again. No. Ever again. Ever again. Well, I'll tie you up with this yarn. You think this is a toy too, huh? Yeah. You think this is a toy? I'm gonna tie you up. I'm gonna tie you up and put you in the kitty stew. Kitty stew pot. I never have to tie up your sister, Trashy. Trashy's a good girl. You're a bad kitty. You're a bad kitty. Look at this mouse. He's gonna go over there. Get him! Oh yeah, great! You did it! Yeah, you did it! Oh, nice! Fling him, kitty, fling him! Fling him, he's so stupid. Fling him, kitty, fling him! Fling him, fling him! Kitty. Show them what you got. I hate these mice. I hate them. I hate them. This is probably a Charles River mouse. Yeah, you eat some tuna too, why not? Oh no! You fell in the tuna, kitty! Oh, you little dum-dum. You're a little dum-dum, aren't you? Oh, you love that toy, but you're probably not playing near it. You're a little dum-dum, aren't you? Careful, kitty. If you get your fur all dirty, then who's going to wash you? She's too young. She's a little kitten. What are you doing? You love that toy. You're a little dumbbell. You're telling me you know quantum physics. You couldn't solve the Schrodinger equation for even the simplest Hamiltonian operators. Schrodinger's cat my ass. Yeah. You got a little spot on your fur. Let me clean it off. Let me clean it off. Okay, good. You're a wild beast. You don't care about a little spot. You're a wild beast. Really get him, take that arrow. Fling him. Fling him, kitty. Wow, what a beast. Stupid mouse. Show him. Yeah, wow, you're such a great contributor to the pack. You're totally not a freeloader. Totally pulling your weight in the family, the pack. Oh, no, I'm not trying to scratch you. I'm not trying to wrestle you. I'm trying to
No, you're stupid. You stupid baby. Why is your toe white? Huh? Why you got a white toe? Oh, he escaped! Ah, get him! He escaped! Oh yeah, fling him good. R.I.P. Mouse. K.O. Fatality. Yeah, have a snack. Have some tuna. I'm worried about her. She hasn't eaten a thing. You just want playtime, huh? She just wants playtime. How about some treats? Treats, treats, treats. Treats. Time for treats. Time for treats, treats. Alert, alert, treats. Alert, alert, alert. Treats, treats, alert. Alert. Treats incoming. Adiabatic computing. Entropy. Turns out entropy is not a problem for this cat. All states. Oh, this stinks. This is stinky. Come here, mama. Come here, mama. She has not yet developed the Pavlovian response. Don't tell me what to do with my cat. My cat's a boss, all right? Make sure you put the string away when you... My cat does whatever she wants, whenever she wants. I take my cat to your place and tell her, poop all over. Go ahead. Do it. Come on, I got some treats for you, stupid. Hey, stupid baby. Oh my God, loves this stupid toy. Right here, kitty, come on. You know what? I'm gonna get you. Come on, get you. I'm gonna get you. I'm gonna get you. I'm gonna get you. Here, come here. Ow! Over here, stupid. Gosh, what a dummy. Over here, over here. Here we go. Okay, here. Now I'll give you some temptations, because I know you like that. I know you like that. Why not? If you don't like temptations, then you're truly a broken cat. Yeah, they're delicious, all right? Even I want one. Isn't delicious? She doesn't even know how to eat it. What kind of cat are you? You're just a mangy, scrawny mutt runt. I'm gonna call you Runt. Yeah, I think her teeth aren't strong enough to... Poor baby. It's a malnourished cat. Teach the cat Albanian? O maca mir. Maca bukur. I don't think she has the teeth to eat these things. She's got like one little tooth. One little tooth. You got one little tooth, don't you? She's leaving little crumbs all over. Oh, you little stupid idiot. Oh, you little stupid idiot.
Well, at least she likes the taste.
At least she's eating something.
Ready? Ready? Let's go!
I'm gonna leave this over here in case you decide to snack. Oh, you want some now? You want some now, stupid? Come here. Eat some of that. Now you're hungry, maybe. Little bozo. You little bozo. You're not ready to be my cat. You're not smart enough to be my cat. All right? Not even close. Look at this idiot. Bumping her head and everything. You're gonna hurt yourself. What the heck? What the heck is that? Alright guys, I got my newest thing. Only real OGs will know what this is. Basically, I'm going to make a lot of new skits with this. It's a device I keep on me at all times. This is a new one. I got my old one in the back. Brand new. You know, some people say don't leave home without it. That's how I feel about my trusty. Who can tell me what this is? Who can tell me what this is? Had one of these since I was a kid. Better than a Glock 17. Way better. Oh, you guys are real noobs. You guys are real noobs. Thank you. Deep fried deals. You get a, you get something, dude. I don't know how to reward you, but you gift a sub? Are you ready a sub? I don't know. This is an HP 12C financial calculator with reverse Polish notation. I keep it on me. Keep it on me, just in case I got to calculate a bond's duration. Had one of these from my Uncle Nick when I was 5 years old. Reverse Polish notation, RPN. That's they strapped. You want to know the yield to maturity of a Ford 2028 bond? Son, you don't know how to work this thing. It doesn't make any sense. Reverse Polish. You want to do 5 or 6 plus 5? What? Even? Doesn't make any sense. Doesn't do anything. Reverse Polish, PV, present value, PMT, FV. I keep it on me at all times in case I need to price an annuity real quick. Hold up. 4% discounted 20 periods. I got your mortgage right here. HP 12C. Right in here. Solar powered? Nah, HP never did a solar power version of the 12C. Come on, man. That's like asking a thoroughbred gangster, did they ever make a Glock 9 milli with a 2% release rate with an extended mag? He's going to say no. In 1988, they switched it up. I know everything about this. HP 12C. 80s bankers with suspenders. You know what I'm talking about. You know what I'm talking about? This is what they used in the Shearson-Lehman bond department, in the Drexel-Burnham trenches. You've been the streets of Philadelphia, or the streets of Wall Street in the 80s. Michael milking these bonds. This is 90s Wall Street. The collared shirt. Look at the CRT monitors. This is what you wore. This, Rolex, suspenders, collared shirt, trading at Bear Stearns. You youngins don't even know what that is. Lehman Brothers didn't even exist. It was First Boston, DLJ, the old school Wall Street places. Just pray you don't meet me in an alleyway and I'm carrying this. I'm holding the 12C. Just pray you don't make me go back to my old me. I don't want to have to discount these cash flows again. Look at this little dummy. She's a little dummy. I thought about naming her Claudia. Like claws, because she's always clawing me too. It doesn't help. You're gonna get tired from running around so much, kitty. Yeah, you are. Yeah, I know. I know. Do you love me, kitty? Do you love me? Do you love me? Do you love me? Do you love me? Now you don't? Okay, sorry. Sorry, Mama. Sorry, Mama. Sorry, Mama. Oh, I thought you loved me. You're too much of a crazy kitten, huh? You're wild. Oh, don't claw me. Stupid. Yeah. Hey, you want to play all day. You want to play all day. I'm too young. I'm too old to play with you. You're too young. You play all day. You want to wrestle. You want to play with the yarn. You want to play with the fish. You never want to eat. You're a little maniac. That's what you are. You're a little maniac. Say hi to everybody. Everybody, hi. Yeah. What? Yeah, you're mad? Oh, you're mad? You don't want to be held? You're not a lap cat, huh? You're a wild cat. Fine. That's fine. Fine. I'll ignore you. I don't need you. I don't need you. Are you stalking and sniffing, looking for that fish? He's over there, stupid. He's over there. Now don't. Yeah. I'll make you into a burrito. Yeah, you'll be a burrito before you know it. Yeah. What kind of meow is that? You don't have the vocal cords to meow with me. I'm not a rapper, so stop rapping at me. I'm not a cat, so stop meowing at me. Everything's her scratching post. Iced coffee. Trashy 2. Trashy's revenge. You're too curious. Yeah, what kind of meow is that? Meow like a real cat. This is just tissues, kitty. You're a stupid kitty. You know that? You're 2 pounds. Useless. Good for nothing. Yeah, I know. Yes, you are. Tell me I should go back to prison. F you too, kitty. Don't talk to me like that. Yeah, well, you know what? This is going to have to be a relationship that we're both going to have to work on. Because you're saying all this bad stuff about me. I'm saying all this bad stuff about you. I'm sorry, kitty. I didn't mean that. I didn't mean that. I didn't mean that. No, I didn't mean that. I'm sorry. Yes, I love you too. Yes, I do. Yes, I love you too. Oh, I'm sorry. I'm sorry I called you a bad kitty. I'm not going to make kitty stew out of you. I'm sorry. Relationships, we got to communicate. Okay, you know what? Fine. I thought we had something, kitty. Made a deal for this kitten straight cash. Upfront balloon payment. 2 milestone payments. Yeah. She said I should have saved my money. Yeah. I know you don't like it here. But I don't like you either. You're just here for the CRISPR experiments. Oh yeah. now you're complaining. Yeah, well, shouldn't have been a bad kitty. I don't want to be CRISPR'd either. Shit. You're about to become Chinese, kitty. Yeah, you cried. Nobody can hear you. I got the mRNA ready. I got it ready. Stupid kitties get the Pfizer vaccine. Not the Pfizer vaccine. Too bad, kitty. Yeah, stop crying then. Stop crying. Stop talking some shit about me. Yo, yeah, you want your sister trashy? She's far away. She can't help you. She can't help you. She forsaken me too. Oh, okay. Now you want to cuddle. Now you want to cuddle. Now you want to cuddle waddle. Okay, now you don't want to cuddle waddle. But I love you. I love you. Cat is wearing a wire. Yeah, she does sound like she's 70 years old. She just don't know how to meow yet. She don't know how to meow yet. She's so small. She's one hand I can carry her. One hand. She's so small. Yeah. Okay, you're mad. I understand you're mad. I understand. I understand you're mad, kitty. We all get mad sometimes. We gotta use this as a learning experience and just grow. Right? Right. I wish I was a young and wild cat too. 9 weeks old, you got your whole life ahead of you, all 9 lives still intact. All you care about is chasing a fishy fish. Fishy fish for your dishy dish. You get a whole tuna feast, you don't even eat it. You don't need it. I don't need it. The mic might be picking up a little purr. Little purr purr. You tired now? You going to sleep? You might be going to sleep. Who loves you? Who loves you? I love you. I love you. I love you. Who loves you? Can we zoom in? Let's zoom in. See if we can't zoom in. I'm sorry I said I'd make stew out of you. I'm sorry I called you a bad kitty. I'm so sorry. I'm so sorry. Will you forgive me? Will you forgive me? I'm so sorry. Yes, I love you too. Yes, I love you too. I'm sorry I called you ugly. But you are pretty ugly. I'm sorry I rubbed your belly. But I'm gonna rub it if I want to. I don't care what you think. You're just a little runt. That's what you are. You're so skinny. Why are you so skinny? She's my favorite. Purr, purr, purr. Purr, purr, purr. Tail wagging. She's camera shy. Camera shy, I think. Black market kitty. That's right. That's right. What's up, kitty? Yeah, what's up?
Oh yeah, I gotta get vert videos, you're right. Alright, let's go back to work. My eyes have rings around them because I was like rubbing them. You still right here? I love you too, kitty. I love you too. I love you too. Allergies are good. She slept with me every night so far. Right in my bed. Right in my bed. All right, now let me eat my food. So I'm accessing this database with a complex, not a complex query, but like a large query. I know there's 90 million, not 90 million, 90,000 records that I'm trying to retrieve. Hold on a second. So this is in a database called Rethink. So I'm trying to get 90,000 records in one query. But I can't tell if like doing it right or not. Yeah, I'm eating some cabbage treats. It's delicious. I'm doing a query to do some deduplication. I'm not a big dog guy or a golf guy.
Yeah, I mean, I know how to play golf. I don't mind it, but I'm a basketball guy. I really want to get this situated. See if I can ask my colleague. All right, let's see. I think I have to call in the big guns and ask my colleague.
Not just to pre-compile and change of opcodes. We're here to sustain and scale all our full nodes.
Proof of stake is hard, but what I do know is when we weren't a shard.
Oh my god.
Don't
That is cringe.
make mistakes or get invalid drunk. See, we freestyle sometimes.
The hell?
Know your gas limit. Stay within it. Don't make mistakes or get invalid drumped. Yo, not just to pre-compile and change of opcodes. We're here to sustain and scale all our full nodes.
Proof of stake is hard, but what I do know is when we weren't a shard, we'll change the whole
Hey!
game though.
E2.0 yo, E2.0 yo, E2.0 yo, E2.0 yo. Shot 3, BLS, Lamport, and all this crypto just sets the stage for Merkle proofs, plasma chains, plat. Seekers knocks coming over, quack. Privacy scaling in a snap. Constant products, oracles, tap. Merkle proofs, plasma chains, plat. Seekers knocks coming over, quack. Privacy scaling in a snap.
Wow. Yeah, that's Vitalik Buterin. I mean, he's pretty great other than the rapping. I guess once a year you have to do something self-deprecating. I don't know.
Constant products, oracles, it's all about the dApps.
Every user, every wallet, every gap. CK miners, double signers, every villain with attacks. Every channel operator, every
Did
black.
he rehearse?
Every phone node, every sneaker, everyone would interact. And that is that.
How would you like to be one of the people dancing?
Constantinople is done. Fork. Plasma group L2 engage. No fork. Color of it coming in waves. Fork. Your boy Carlos is from New York.
Wow. Maybe I'm glad I didn't go. I use the Philco Magic Touch, and then I also have an Alienware keyboard, both plugged in. I did rebuy Philco. My old one is too old. Of course I know Albanian. Do you want me to... Do you want to do a type racer? Let's do type racer. I'm just going to
to put this away real quick. Where'd that stupid kitty go? Oh, hey! Hey, I love you. She's in the corner. You're in the corner. Why are you in the corner? You're in with all the dust. She's a dirty kitty. Alright, let's do type racer. I don't know what account. Do I have to do my old account? Maybe I can log into that. Let's see. Is the idea of God in mathematics? Yeah, to some extent. I would say when you look at prime numbers, I see God in prime numbers. Maybe that's corny. Oh, I should upgrade to premium. I do still have MP3s of the Wu-Tang album. Yeah, I have unreleased music from everyone. That's a lot of my hobbies for a little while. I don't think you have to have a big ego to work on Wall Street. In fact, I think people with the smaller egos Alright, so I guess we're racing in 10 seconds, so join up if you want to play. Three, two, one. I came in third. I haven't used this keyboard. That was a weak performance. I'm not sure I can do 150, but we'll try again. 10 seconds. Oh, shit. Oh my god, what's going on here? This doesn't count.
Oh shoot, I have to call a friend of mine who I haven't been trying to blow off, but I forgot to call. Alright guys, I should run. Great stream today. Really had a lot of fun hanging out with everyone. I'll catch y'all soon.